14. Chapter 3. Principles of crop production. Learning objectives. After studying this chapter, students will be able to know meaning of climate and its major components. What is soil and its role in crop production? What are the important plant nutrients and their role? What is crop and basis of their classification? Meaning of farming system and types of farming systems. Role of water in crop production and their management. Climate. The weather conditions prevailing in an area in general or over a long period is called climate. It is a measure of the average pattern of variation in temperature, humidity, atmospheric pressure, wind, precipitation, atmospheric pressure and other meteorological variables in a given region over long periods of time. The climatic factors include rainfall and water, light, temperature, relative humidity, air, and wind. They are abiotic components, including topography and soil, of the environmental factors that influence plant growth and development. Influence of climate on crops. Climate is the most important dominating factor influencing the suitability of a crop to a particular region. The yield potential of the crop mainly depends on climate. More than 50% of variation of crops is determined by climate. The most important climatic factors that influence growth, development and yield of crops are solar radiation, light, temperature and rainfall. Solar radiation, sunlight. Solar radiation, sunlight, is that visible portion of the solar radiation or electromagnetic spectrum. It is a form of kinetic energy that comes from the sun in tiny particles called quanta or photons, traveling in waves. Sunlight should be carefully considered when selecting plants for your garden. All plants need sunlight for photosynthesis. Photosynthesis is the process by which light, chlorophyll, energy, carbon dioxide and water interact within the plant to produce the sugar and starches it needs to survive. All plants require some amount of sunlight. Good Introductory Agriculture. 15. Some need many hours of full sun while others do best largely in the shade. Plants need sunlight in order to survive and, without sunlight, or artificial light if grown indoors, plants would not grow strong and healthy and would die, or at best, be very unhealthy. Three properties of light affect plant growth and development are light quality, light intensity, and day length or photoparide. Light quality refers to the specific wavelength of light. Light intensity is the degree of brightness that a plant receives, and day length is the duration of the day with respect to the light period. Temperature. Temperature is the degree of hotness or coldness of a substance is called temperature. It is commonly expressed in degree Celsius or centigrade, 0 C, and degree Fahrenheit, 0 F. Temperature influences growth processes of all plants such as photosynthesis, respiration, transpiration, breaking of seed dormancy, seed germination, protein synthesis, and translocation. At high temperature the translocation of photosynthesis is faster so plants tend to mature earlier. In general, plants survive within a temperature range of 0 to 50 C. The favorable or optimal day and night temperature range for plant growth and maximum yield varies among crop species. Enzyme activity and the rate of most chemical reactions generally increase with rise in temperature. But at excessively high temperatures, denaturation of enzymes and other proteins occur. Excessively low temperature can also cause limiting effects on plant growth and development. For example, water absorption is inhibited when the soil temperature is low because water is more viscous at low temperatures and less mobile, and the protoplasm less permeable. At temperatures below the freezing point of water, there is change in form of water from liquid to solid. The expansion of water as it solidifies in living cells causes the rupture of the cell walls. Air. The air is a mixture of gases in the atmosphere. About 75% of this air is found in the troposphere. The oxygen and carbon dioxide in the air is important to the physiology of plants. Oxygen is essential in respiration for the production of energy that is utilized in various growth and development processes. Carbon dioxide is a raw material in photosynthesis. Wind. Air movement or wind is due to the existence of pressure gradient on a global or local scale caused by differences in heating. On a global scale, it consists of the jet stream flow and movement of large air masses. On the local scale, only a smaller quantity of air moves. Surface winds are lower and less turbulent at night due to the absence of solar heating. When air that is close to the ground cools, it contracts and the pressure rises. When it warms, it expands and loses pressure. Where both cold and warm air occur in proximity, as over 16. Lake and its adjacent shore, the cold flows to the direction of the warm air or from high to low pressure area to correct the pressure imbalance. This also happens in tropical Asia but in a larger and more complex way, as the monsoon winds. This climatic factor serves as a vector of pollen from one flower to another, thus aiding in the process of pollination. It is therefore essential in the development of fruit and seed from wind-pollinated flowers as in many grasses. Moderate winds favor gas exchanges, but strong winds can cause excessive water loss through transpiration as well as lodging or toppling of plants. When transpiration rate exceeds that of water absorption, partial or complete closure of the stomater may ensue which will restrict the diffusion of carbon dioxide into the leaves. As a result, there will be a decrease in the rate of photosynthesis, growth and yield. Edmund et al. 1978. Each of the above discussed climatic factors has been shown to produce limiting effects on various growth processes. However, the various climatic factors always operate together and interact with each other under natural conditions. Rainfall, precipitation. Rainfall is the most common form of precipitation. It is the falling of water in droplets on the surface of the earth from clouds. Other forms of precipitation are freezing rain, sleet or ice pellets, snowfall, and hail. The amount and regularity of rainfall vary with location and climate types and affect the dominance of certain types of vegetation as well as crop growth and yield. Relative humidity RH. Relative humidity RH is the amount of water vapor in the air, expressed as the proportion, in percent, of the maximum amount of water vapor it can hold at certain temperature. For example, an air having a relative humidity of 60% at 27 degrees Celsius temperature means that every kilogram of the air contains 60% of the maximum amount of water that it can hold at that temperature. The relative humidity affects the opening and closing of the stomater which regulates loss of water from the plant through transpiration as well as photosynthesis. A substantial understanding of this climatic factor is likewise important in plant propagation. Newly collected plant cuttings and bare root seedlings are protected against desiccation by enclosing them in a sealed plastic bag. The propagation chamber and plastic tent are also commonly used in propagating stem and leaf cuttings to ensure a condition with high relative humidity. Soil. Soil is a mixture of broken rocks and minerals, living organisms, and decaying organic matter called humus. Humus is dark, soft and rich in nutrients. Soil also includes air and water. Introductory Agriculture. 17. Organisms in the soil need air and water to survive. Having these essential materials, air, water, and organic matter, makes it possible for plants, bacteria, fungi and small animals like earthworms and insects to live in the soil. All the living things in the soil, plus essential materials that these organisms use to survive, form the soil ecosystem. Scientists study the soil ecosystem because they want to understand how organisms relate to one another and to the environment that surrounds them. Soil generally consists of visually and texturally distinct layers, also called profiles, which can be summarized as follows from top to bottom. Figure. Photograph of typical soil profile. O. Organic matter, superficial organic deposit with little layer of plant residues in relatively non-decomposed form. A. Surface soil, organics mixed with mineral matter. The layer of mineral soil with the most organic matter accumulation and soil life. This layer alluviates, is depleted of iron, clay, aluminum, organic compounds, and other soluble constituents. When alluviation is pronounced, a lighter colored A subsurface soil horizon is apparent at the base of the A horizon. A horizons may also be the result of a combination of soil bioturbation and surface processes that winnow fine particles from biologically mound topsoil. In this case, the A horizon is regarded as a biomantle. Bedrock. Horizons. 18. B. Subsoil. Subsurface layer reflecting chemical or physical alteration of parent material. This layer accumulates iron, clay, aluminum and organic compounds, a process referred to as alluviation. C. Parent rock. The parent material in sedimentary deposits. Layer of large unbroken rocks. This layer may accumulate the more soluble compounds. R. Bedrock. The parent material in bedrock landscapes. This layer denotes the layer of partially weathered bedrock at the base of the soil profile. Unlike the above layers, R horizons largely comprise continuous masses of hard rock that cannot be excavated by hand. Soils formed in situ will exhibit strong similarities to this bedrock layer. These areas of bedrock are under 50 feet of the other profiles. Indian Standard Soil Classification System. Classification based on grain, soil particle, soil separates, size. The range of particle sizes encountered in soils is very large, from boulders with dimension of over 300 mm down to clay particles that are less than 0.002 mm. Some clay contains particles less than 0.001 mm in size which behave as colloids, i.e. do not settle in water. In the Indian Standard Soil Classification System, ISSCS, soils are classified into groups according to size, and the groups are further divided into coarse, medium and fine subgroups. The grain size range is used as the basis for grouping soil particles into boulder, cobble, gravel, sand, silt or clay. Very coarse soils. Boulder size. Greater than 300 mm. Cobble size. 80 to 300 mm. Coarse soils. Gravel size, G. Coarse. 20 to 80 mm. Fine. 4. 75 to 20 mm. Sand size, S. Coarse. 2 to 4.75 mm. Medium. 0. 425 to 2 mm. Fine. 0. 075 0.425 mm. Fine soils. Silt size, M. 0. 002 0.075 mm. Clay size, C. Less than 0.002 mm. Gravel, sand, silt, and clay are represented by group symbols G, S, M, and C respectively. Physical weathering produces very coarse and coarse soils. Chemical weathering produces generally fine soils. Soil separates are specific ranges of particle sizes. In the United States, the smallest particles are clay particles and are classified by the USDA as having diameters of Introductory Agriculture. 19. Less than 0.002 mm. The next smallest particles are silt particles and have diameters between 0.002 mm and 0.05 mm. The largest particles are sand particles and are larger than 0.05 mm in diameter. Soil texture. Soil texture is the relative proportions of sand, silt, or clay in a soil. Soil texture is a qualitative classification tool used in both the field and laboratory to determine classes for agricultural soils based on their physical texture. The classes are distinguished in the field by the textural field, which can be further clarified by separating the relative proportions of sand, silt and clay using grading sieves, the particle size distribution, PSD. The class is then used to determine crop suitability and to approximate the soil's responses to environmental and management conditions such as drought or calcium, lime, requirements. A qualitative rather than a quantitative tool, it is a fast, simple and effective means to assess a soil's physical characteristics. Textural triangle. The soil textural class is a grouping of soils based upon these relative proportions. Soils with the finest texture are called clay soils, while soils with the coarsest texture are called sands. However, a soil that has a relatively even mixture of sand, silt, and clay and exhibits the properties from each separate is called a loam. There are different types of loams, based upon which soil separate is most abundantly present. If the percentages of clay, silt, and sand in a soil are known, primarily through laboratory analysis, you may use the textural triangle to determine the texture class of your soil. 100, 90, 80, 70, 60, 50, 40, 30, 100, 90, 80, 70, 50, 40, 8, 6, 50. Clay Importance of clay and other particles of similar size. Clay particles, as well as other particles of similar size, are important components of a soil. There is a fundamental difference between soils that contain large amounts of sand particles and soils that contain large amounts of very small particles, such as clay. That difference is surface area. The total surface area of a given mass of clay is more than 1,000 times the total surface area of sand particles with the same mass. To put this idea into perspective, imagine a single cube with 6 sides. This cube represents a sand particle. Now, imagine that you break the single cube up into 100 smaller cubes, which represent 100 clay particles. These 100 cubes each have 6 sides. Essentially, by breaking up the larger cube, you have exposed many more surfaces. Thus, the total surface area of the smaller cubes will be much greater than the surface area of the single cube. This increase in surface area has an important implication in nutrient management because it provides many places for soil particles to retain and supply nutrients such as calcium, potassium, magnesium, phosphate, and water for plant uptake. Types of very small particles within the soil. The most common clay minerals in Maui soil are called layered silicate clays or phyllosilicates. There are different types of layered silicates such as kaolinite, halloysite, montmorillonite, and vermiculite. The various types of layered silicates differ greatly, as we will discuss later. Amorphous minerals such as allophane, amogolite, and ferrihydride may be found in the volcanic soils of Hawaii that developed from volcanic ash. Like silicate clays, these minerals have a very high surface area. As a result, soils with amorphous minerals hold large amounts of water and stored nutrients, depending on the degree of weathering. Aluminum and iron oxides are typically found in the highly weathered soils of the tropics. As clay minerals are intensely weathered, the structure of silicates clays change. Particularly, the silicate clays lose silica. What remains in the soil are aluminum and iron oxides. Gypsite is an example of an aluminum oxide, which has a grayish, whitish hue. Goethite is an example of an iron oxide, which imparts a reddish color to the soil. Properties of oxides. Oxides are fairly stable and resistant to further weathering. Oxides can act like a glue and hold other soil particles together. Oxides can tie up nutrients, such as phosphorus. Oxides have a high anion exchange capacity, ACE. Humus is the portion of organic matter that is mostly resistant to decomposition and remains in the soil. Humus is composed of small particles, with tremendous surface area. These particles have a very great capacity to retain and supply nutrients, as well as hold water. Introductory Agriculture. 21. Soil structure. Soil structure is the arrangement of soil particles into groupings. These groupings are called beds or aggregates, which often form distinctive shapes typically found within certain soil horizons. For example, granular soil particles are characteristic of the surface horizon. Soil aggregation is an important indicator of the workability of the soil. Soils that are well aggregated are said to have good soil tilth. The various types of soil structures are provided in figure. Granular resembles cookie crumbs and is usually less than 0.5 cm in diameter. Commonly found in surface horizons where roots have been growing. Prismatic, vertical columns of soil that might be a number of cm long. Usually found in lower horizons. Blocky, irregular blocks that are usually 1.5, 5.0 cm in diameter. Single-grained, soil is broken into individual particles that do not stick together. Always accompanies a loose consistence commonly found in sandy soils. Laty, thin, flat plates of soil that lie horizontally. Usually found in compacted soil. Columnar, vertical, columns of soil that have a salt cap at the top. Found in soils of climates. Figure, types of soil structures in soils. Source. 22. Soil aggregates. Generally, only the very small particles form aggregates, which include silicate clays, volcanic ash minerals, organic matter, and oxides. There are various mechanisms of soil aggregation. Mechanisms of soil aggregation. Soil microorganisms excrete substances that act as cementing agents and bind soil particles together. Fungi have filaments, called hyphae, which extend into the soil and tie soil particles together. Roots also excrete sugars into the soil that help bind minerals. Oxides also act as glue and join particles together. This aggregation process is very common to many highly weathered tropical soils and is especially prevalent in Hawaii. Finally, soil particles may naturally be attracted one another through electrostatic forces, much like the attraction between air and a balloon. Aggregate stability. Stable soil aggregation is a very valuable property of productive soils. Yet, the stability of soil aggregation is very reliant on the type of minerals present in the soil. Certain clay minerals form very stable aggregates, while other clay minerals form weak aggregates that fall apart very easily. Highly weathered silicate clays, oxides, and amorphous volcanic materials tend to form the most stable aggregates. The presence of organic matter with these materials improves stable aggregate formation. In nutrient management, the aggregate stability is important because well-aggregated minerals are well-drained and quite workable. In contrast, less weathered silicate clays, such as montmorillonite, form weak aggregates. Some silicate clays are said to have a shrink-swell potential. This means that the soil minerals expand, or swell, when wet, causing the soil to become sticky and drain poorly. When dry, these soils shrink and form cracks. The makeup of the lattice structure of silicate clays determines the shrink-swell potential. Although there are no soils with a shrink-swell potential in Maui, these soils may be found on Molokai. Tillage. Tillage is the agricultural preparation of soil by mechanical agitation of various types, such as digging, stirring, and overturning. Examples of human-powered tilling methods using hand tools include shoveling, picking, mattock work, hoeing, and raking. Examples of draft animal introductory agriculture. 23. Powered or mechanized work include plowing, overturning with mold boards or chiseling with chisel shanks, rototilling, rolling with cultipackers or other rollers, harrowing, and cultivating with cultivator shanks teeth. Small-scale gardening and farming, for household food production or small business production, tends to use the smaller-scale methods above, whereas medium to large-scale farming tends to use the larger-scale methods. There is a fluid continuum, however. Any type of gardening or farming, but especially larger-scale commercial types, may also use low-till or no-till methods as well. Tillage can also mean the land that is tilled. The word cultivation has several senses that overlap substantially with those of tillage. In a general context, both can refer to agriculture. Within agriculture, both can refer to any of the kinds of soil agitation described above. Additionally, cultivation, or cultivating, may refer to an even narrower sense of shallow, selective secondary tillage of row crop fields that kills weeds while sparing the crop plants. Tillage is often classified into two types, primary and secondary. There is no strict boundary between them so much as a loose distinction between tillage that is deeper and more thorough, primary, and tillage that is shallower and sometimes more selective of location, secondary. Primary tillage such as plowing tends to produce a rough surface finish, whereas secondary tillage tends to produce a smoother surface finish, such as that required to make a good seed bed for many crops. Harrowing and rototilling often combine primary and secondary tillage into one operation. It can be defined as primary tillage loosens the soil and mixes in fertilizer and or plant material, resulting in soil with a rough texture. Secondary tillage produces finer soil and sometimes shapes the rows, preparing the seed bed. It also provides weed control throughout the growing season during the maturation of the crop plants, unless such weed control is instead achieved with low-till or no-till methods involving herbicides. The seed bed preparation can be done with harrows, of which there are many types and subtypes: dibbles, hoes, shovels, rotary tillers, subsoilers, ridge or bed forming tillers, rollers, or cultivators. The weed control, to the extent that it is done via tillage, is usually achieved with cultivators or hoes, which disturb the top few centimeters of soil around the crop plants but with minimal disturbance of the crop plants themselves. The tillage kills the weeds via two mechanisms: uprooting them, burying their leaves, cutting off their photosynthesis, or a combination of both. Weed control both prevents the crop plants from being outcompeted by the weeds for water and sunlight, and prevents the weeds from reaching their seed stage, thus reducing future weed population aggressiveness. 24. Tillage systems. There are different types of tillage systems. Reduced tillage. Reduced tillage leaves between 15 and 30% residue cover on the soil of 500 to 1,000 pounds per acre, 560 to 1,100 kg per hectare of small grain residue during the critical erosion period. This may involve the use of a chisel plow, field cultivators, or other implements. Intensive tillage. Intensive tillage leaves less than 15% crop residue cover or less than 500 pounds per acre, 560 kg per hectare of small grain residue. This type of tillage is often referred to as conventional tillage, but as conservational tillage is now more widely used than intensive tillage. In the United States, it is often not appropriate to refer to this type of tillage as conventional. Intensive tillage often involves multiple operations with implements such as a mold board, disc, and or chisel plow. Then a finisher with a harrow, rolling basket, and cutter can be used to prepare the seed bed. There are many variations. Conservation tillage. Conservation tillage leaves at least 30% of crop residue on the soil surface, or at least 1,000 pounds AC, 1,100 kg per hectare of small grain residue on the surface during the critical soil erosion period. This slows water movement, which reduces the amount of soil erosion. Conservation tillage also benefits farmers by reducing fuel consumption and soil compaction. By reducing the number of times the farmer travels over the field, farmers realize significant savings in fuel and labor. In most years since 1997, conservation tillage was used in US cropland more than intensive or reduced tillage. However, conservation tillage delays warming of the soil due to the reduction of dark earth exposure to the warmth of the spring sun, thus delaying the planting of the next year's spring crop of corn. No-till, never use a plow, disc, etc. ever again. Aims for 100% ground cover. Strip till, narrow strips are tilled where seeds will be planted, leaving the soil in between the rows untilled. Mulch till, soil is covered with mulch to conserve heat and moisture. Rotational tillage, tilling the soil every 2 years or less often, every other year, or every third year, etc. Ridge till, ridges are made with the help of ridge maker for sowing or planting. Effects of tillage. Positive effects of tillage. Plowing. Loosens and aerates the top layer of soil, which facilitates planting the crop. Introductory agriculture. 25. Helps mix harvest residue, organic matter, humus, and nutrients evenly into the soil. Mechanically destroys weeds. Dries the soil before seeding. In wetter climates, tillage aids in keeping the soil drier. When done in autumn, helps exposed soil crumble over winter through frosting and defrosting, which helps prepare a smooth surface for spring planting. Negative effects of tillage. Dries the soil before seeding. Soil loses a lot of nutrients, like nitrogen and fertilizer, and its ability to store water. Decreases the water infiltration rate of soil. Results in more runoff and erosion since the soil absorbs water slower than before. Tilling the soil results in dislodging the cohesiveness of the soil particles, thereby inducing erosion. Chemical runoff. Reduces organic matter in the soil. Reduces microbes, earthworms, ants, etc. Destroys soil aggregates. Compaction of the soil, also known as a tillage pan. Eutrophication, nutrient runoff into a body of water. Can attract slugs, cutworms, armyworms, and other harmful insects to the leftover residues. Crop diseases can be harbored in surface residues. Alternatives to tilling. Modern agricultural science has greatly reduced the use of tillage. Crops can be grown for several years without any tillage through the use of herbicides to control weeds, crop varieties that tolerate packed soil, and equipment that can plant seeds or fumigate the soil without really digging it up. This practice, called no-till farming, reduces costs and environmental change by reducing soil erosion and diesel fuel usage. Researchers are investigating farming in polyculture that would eliminate the need for both tillage and pesticide, such as no-dig gardening. Erosion. Soil is one of the most important natural resources of man. Soils are essential for man for growing crops, fodder and timber. Once the fertile portion of the earth's surface is lost, it is 26. Very difficult to replace it. In India, the destruction of the topsoil has already reached an alarming proportion. Land degradation problems have resulted in increasing depletion of the productivity of the basic land stock through nutrient deficiencies. In addition to the direct loss of crop producing capacity, soil erosion increases the destructiveness of floods and decreases the storage capacity of water in reservoirs. It is, therefore, necessary to keep soil in place and in a state favorable to its highest productive capacity. Soil erosion. The process of destruction of soil and the removal of the destroyed soil material constitutes soil erosion. The vastly accelerated process of soil removal brought about by the human interference with the normal disequilibrium between soil building and soil removal is designated as soil erosion. Soil erosion takes place through two agencies, water and air, and called as water erosion and wind erosion. Types of water erosion. Erosion of soil by water is quite significant and takes place chiefly in two ways: a. Sheet erosion, b. Gully erosion. A. Sheet erosion. Sheet movement of water causes sheet erosion and depends on the velocity and quantity of pronounced surface runoff and the erodibility of the soil itself. In such cases, the soil is eroded as layers from the hill slopes, sometimes slowly and insidiously and sometimes more rapidly. Sheet erosion is more or less universal on all bare fallow land, all uncultivated land whose plant cover has been thinned out by overgrazing, fire or other misuse, and all sloping cultivated fields and on sloping forest, scrub jungles where natural porosity of soil has been removed by heavy grazing, felling of trees or burning etc. The particles loosened and shifted by the raindrops are carried down slope by a very thin sheet of water which moves along the surface. The impacts of the raindrops increases the turbulence and transporting capacity of this and channelized sheet wash, which results in the uniform skimming of the topsoil. Sheet erosion is considered as dangerous as it may continue for years but may or may not leave any trace of the damage. Sheet erosion is common in the Himalayan foothills, in Assam, Western Ghats and Eastern Ghats. Introductory Agriculture. 27. When sheet erosion continues unchecked, the silt-laden runoff forms well-defined minute finger-shaped grooves over the entire field. Such thin channeling is known as rill erosion, which is active over wide areas in Bihar, Uttar Pradesh, Madhya Pradesh and in semi-arid areas of Maharashtra, Karnataka, Andhra Pradesh and Tamil Nadu. B. Gully erosion. On a gentle slope, adequately covered by vegetation, clay soil will resist erosion to a great extent and the water forms small rivulets which can then erode deeper. The rivulets in turn join together to form larger channels until gullies are formed. Gradually deep gullies cut into the soil and then spread and grow until all the soil is removable from the sloping ground. This phenomenon, once started and if not checked, goes on extending and ultimately the whole land is converted into a bad land topography. Gully erosion is more common in areas where the river system has cut down into elevated plateaus so that feeders and branches carve out an intricate pattern of gullies. Apart from this, it also takes place in relatively level country whenever large blocks of cultivation give rise to concentration of field runoff. Wind erosion. It occurs in dry climatic areas having a sparse and low vegetation cover on mechanically weathered, loosened surficial material. Dust storms are the principal agents of wind erosion. The topsoil is often blown off from the surface, rendering it infertile. Besides, with the decrease in the wind velocity cause sand particles get deposited in some areas covering the existing soil and rendering it unproductive. Soil conservation. Soil conservation is the prevention of soil from erosion. It includes all such measures which help in protecting the soil through contour terracing and bunding, construction of bunds across gullies, leveling of uneven land and raising grass and other vegetation on land are the small measures which are usually taken by farmers to protect soil from erosion. Such methods are quite effective in areas where the degree of erosion is not serious, as in the semi-arid tracts of the peninsula and part of Gujarat, Madhya Pradesh, Uttar Pradesh, Haryana and Punjab. In the tropical forest areas, shifting cultivation known as jumming in Assam, Punam in Kerala, Podu in Andhra Pradesh and Orissa. Methods of soil conservation. Contour bunding is the construction of small bunds across the slopes of the land on a contour. If the plowing is done at right angles to the hill slope, following the natural contour of the hill, the ridges and furrows break the flow of the water down the hill. This prevents excessive soil loss, as gullies are likely to develop and also reduce runoff so plants 28. Receive more water. Row crops and small grains are often planted in contour patterns so that the plants can absorb much of the rain and erosion is minimized. Terracing. Slopes may be cut into a series of terraces with sufficient level ground on each terrace for cultivation, and an outer wall at the edge to retain the soil and to slow down the flow of rainwater down the slope. Terracing is widely used in monsoon Asia for wet paddy cultivation, as the excess water and silt can be retained at each terrace to form flooded paddy fields. Terraces are also used in temperate and semi-arid regions where slopes are steep. Terracing enables farmers in mountainous regions to utilize the steep ground on the favored sunny slopes of valleys for vines or other crops. Strip cropping. Crops may be cultivated in alternate strips, parallel to one another. Some strips may be allowed to lie fallow while others are sown to different kind of crops, e.g. grains, legumes, small tree crops. The various crops ripen at different times of the year and harvested at intervals. This ensures that at no time will the entire area be left bare or exposed. The tall growing crops act as windbreaks in the strips, which are often parallel to the contours, help to increase water absorption by the soil by slowing down runoff. Fallowing. Sometimes it is important to allow much used land to rest or lie fallow, so that the natural forces can act on the soil. The decayed natural vegetative matter helps to increase the plant nutrients in the soil. Fallowing also increases the subsoil moisture and improves the general structure of the soil. Winter fallow is commonly practiced in temperate regions after the harvest, but cultivation is resumed in the spring after the snow and frost have weathered the topsoil. Long periods of fallow cannot be allowed, however, in intensively run farms as farmers cannot afford it. Cover cropping. In some cases, as in plantations, where the gestation period of tree crops is long, cover crops may be interplanted between the young trees. Creepers are preferred because they spread around and form a useful cover that protects the topsoil from the full force of the tropical downpours. Care must be taken that cover crop does not compete with the young trees for the essential plant nutrients, and leguminous crops are often used because they add nitrogen to the soil. Cover crops may be grown simply to protect the soil or may consist of other valuable plants such as vegetables which provide an income while the plantation crops matures. Some such crops, e.g. cotton, maize or tobacco should be avoided because they exhaust the soil or promote soil erosion instead of preventing it. Crop rotation. It is not advisable to grow the same crop in the same field for more than 2 years in succession as the crop will tend to exhaust one particular kind of mineral nutrient. For example, potatoes require much potash, but wheat requires nitrates. Thus, it is best to alternate crops in the fields. Legumes such as peas, beans, clover, vetch and many other plants add nitrates to the soil by converting free nitrogen in the air into nitrogenous nodules on their roots. Thus, if they are included in the crop rotation, nitrogenous fertilizer can be dispensed with. By rotating different types of crops in successive years, soil fertility can be naturally maintained. Introductory Agriculture. 29. Crop diversification. This practice is often like crop rotation in that it helps to maintain soil fertility. Where annually harvested crops are grown, they can be alternated in the field. Where perennial crops like tree crops are grown, however, the chief importance of crop diversification to the farmer is economic. In particular, it reduces the danger of depending on a single crop, monoculture. Control of gully erosion. Gully control by the diversion of runoff proved to be an economical measure. The best method of controlling existing gullies is to re-establish vegetation. Two of the most important types are the sod strip eroded earth till. Windbreak. The principal method of reducing surface velocity of wind upon which would depend the abrasive and transportation capacity are vegetal measures. Soil fertility and productivity. Soil fertility. Soil fertility is the ability of the soil to provide all essential plant nutrients in available forms and in a suitable balance. Soil productivity. The capability of soil to produce specified crop yield under well-defined and specified systems of management of inputs and environmental conditions. Factors governing soil fertility. Parent material. Fertility of a soil depends on the chemical composition of parent material from which it derived. Soils on the upper slope are less fertile than the soils on lower slope because high leaching and erosion on upper slope. Climate. In tropical climate, decomposition of organic matter is faster than temperate climate. The soils of tropical regions are less fertile when compared to temperate region. Depth of soil profile. Deep soils are more fertile than the shallow soils and the roots are spread well enough in deep soils than the shallow soils. Physical condition of soil. The soil texture and soil structure influence the soil fertility. Artificial factors. I. Water logging E. Cropping system E. Toxic chemicals and pesticide in the soil. 30. Difference between soil fertility and productivity. Soil fertility. Soil productivity 1. It is an index of available nutrient to plants 1. It is used to indicate crop yields. 2. Influenced by the physical, chemical and biological factors of the soil. 2. Depends upon fertility and location. 3. It is the function of available nutrients of the soil. 3. It is the function of soil fertility, management and climate. 4. All fertile soils are not productive. 4. All productive soils are fertile. 5. It is an inherent property of the soil. 5. It is not the inherent property of the soil. What is soil organic matter? Soil organic matter, SOM, is mainly composed of carbon, hydrogen and oxygen but also has small amounts of nutrients such as nitrogen, phosphorus, sulfur, potassium, calcium and magnesium contained within organic residues. It is divided into living and dead components and can range from very recent inputs such as stubble to largely decayed materials that are thousands of years old. Less than 15% of below ground soil organic matter such as roots, fauna and microorganisms as living figure. Soil organic matter also exists as 4 distinct fractions which vary widely in their size, composition and turnover times in the soil table. 1. Dissolved organic matter 2. Particulate organic matter 3. Humus 4. Resistant organic matter. Introductory agriculture. Introductory agriculture. 31. Living organic matter. Particulate organic matter. Fresh residues. Living organisms. Humus. Resistant organic matter. 10%. 10%. 15%. 65%. Figure. The vast majority of soil organic matter is dead or decaying with living organisms making up less than 15% of the soil organic matter pool. Table. The size, turnover rate and composition of the 4 soil organic matter fractions. Fraction size micrometers, micro m and millimeters mm. Turnover time. Composition. Dissolved organic matter. Less than 45 micrometers in solution, minutes to days soluble root exudates, simple sugars and decomposition byproducts. It generally makes up less than 5% of total soil organic matter. Particulate organic matter. 53 micrometers minus 2 millimeters. 2 to 50 years fresh or decomposing plant and animal matter with identifiable cell structure. Makes up between 2 to 25% of total soil organic matter. Humus. Less than 53 micrometers. Decadal, 0 years. Older, decayed organic compounds that have resisted decomposition. Can make up more than 50% of total soil organic matter. Resistant organic matter. Less than 53 micrometers, less than 2 millimeters 0 years. Relatively inert materials such as chemically resistant materials or organic remnants, e.g. charcoal. Can be up to 10% of soil organic matter. 32. Storing the carbon contained in organic matter within the soil is seen as one way to mitigate climate change by reducing greenhouse gas emissions, in this case carbon dioxide, but to do this an increase in the most stable or resistant fractions of organic matter is required. How is soil organic matter different to soil organic carbon? Soil organic carbon, SOC, is different to organic matter because it refers only to the carbon component of organic compounds. Soil organic matter is difficult to measure directly, so laboratories tend to measure and report soil organic carbon. A conversion factor is available to report soil organic matter when required. About 58% of the mass of organic matter exists as carbon. So if we know the organic carbon content in a soil, we can estimate the amount of organic matter. Organic matter percent equals total organic carbon percent by 1.72. While this ratio can vary in different soils, using a conversion factor of 1.72 provides a reasonable estimate of soil organic matter and is suitable for most purposes. What is humus? Before to define humus, let's look at some similar terms that add confusion to the story. Humus layer. This term is sometimes used to describe an upper level of soil, that dark black layer, such as in that humusy layer of soil. Although the dark color is probably due to humus, humus is not a layer in soil. There is no such thing as a humus layer. Humus soil. This term is floated around the net and it is not clear what it means. Is it soil with humus in it? Most soil has some humus, so why not just call it soil? It is a term that should not be used. Humus equals compost. In agriculture and gardening, the term humus is sometimes used to describe well-aged compost. You can buy bags of stuff labeled humus at gardening centers, but this is just mislabeled compost. This is an incorrect use of the term. Compost is plant material that is slightly decomposed. Even aged, well-rotted compost is still only slightly decomposed. Once added to soil, compost will continue to decompose for several years. Compost is not humus. Fulvic acid, humic acid and human, these are terms referring to different subparts of humus. They have specific scientific definitions and should not be used in place of the word humus. From the point of view of gardeners, these terms should not be used. Humification, the process of creating humus. The best way to understand humus is to understand how it is formed. Dead plant and animal material consist of organic matter. Organic matter is a catch-all phrase used to describe a wide range of molecules including starches, proteins, sugars, carbohydrates, amino acids, etc. When organic matter starts to decompose, these molecules are broken down into smaller introductory agriculture. 33. And smaller molecules by the microorganisms in the soil, mostly bacteria and fungi. This is a complex process. Most of the usable chemicals in the organic matter are extracted by the microorganisms and are eventually made available to plants. At some point, all of the good stuff in the organic matter is used up and some molecules remain that can't be used by microorganisms or plants. This remaining material is called humus. It consists mostly of carbon and so it is still organic, but microorganisms just can decompose it any further. Humus is so stable that it can persist in the soil for hundreds of years. Humus consists of very large complex carbon-based molecules. More recent research suggests that it might actually consist of smaller molecules that are conglomerated into large complex systems. Scientists still don't understand humus completely. Humus is very important for soil. The high amount of humus is good for soil. Breakdown of organic matter. Organic matter breakdown is not a single chemical transformation but a complex process. Breakdown of organic matter involves chemical alteration of organic matter, physical fragmentation and finally release of mineral nutrients. Organic matter breakdown is a biological process because it is the soil organisms, microorganisms, earthworms, microtheropods, ants, beetles, etc. that perform the chemical and physical changes. Different organisms are involved with the different stages of these processes. Breakdown starts almost immediately after the organism, or part of it, dies. The organic material is colonized by microorganisms that use enzymes to oxidize the organic matter to obtain energy and C. For leaves and roots, their surfaces are colonized by microorganisms even before they die. Soil animals such as earthworms assist in the decomposition of organic matter by incorporating it into the soil where conditions are more favorable for decomposition than on the surface. Earthworms and other larger soil animals, such as mites, columbola and ants, fragment organic material increasing the surface area and allowing more microorganisms to colonize the organic matter and decompose it. During decomposition, the organic molecules in organic matter are broken down into simpler organic molecules that require further decomposition or into mineralized nutrients. The compounds in organic matter vary in the ease with which microorganisms can break them down. The first organic compounds to be broken down are those that are easy to break down, including amino acids and sugars. Cellulose will break down more slowly and lignin, phenols and waxes will remain in the soil for the longest time. Why do stems and leaves decompose at different rates? Stems and leaves decompose at different rates because they are made up of different kinds of molecules that require different enzymes for their degradation. Leaves generally have more cellulose than lignin. Stems generally have more lignin than leaves. Cellulose is a simpler 34 molecule which is able to be decomposed by microorganisms more quickly than lignin. Cellulose tends to be a straight, chain-like molecule. Lignin has a complex, folded structure so the enzymes cannot break up the component parts quickly. When lignin is linked within the plant cell walls with cellulose, it makes it harder to degrade the cellulose. After 10 weeks, most parts of leaves will have been degraded, but it may take about 30 weeks to degrade the same amount of stem material. Mineralization of organic matter. This needs to be considered in terms of carbon, nitrogen, phosphorus and sulfur. Mineralization is the biological process where organic compounds in organic matter are chemically converted by the microorganisms in soil to simpler organic compounds, other organic compounds or mineralized nutrients. Bacteria and fungi are responsible for most of the mineralization of organic matter in soils. Microorganisms release enzymes that oxidize the organic compounds in organic matter. The oxidation reaction releases energy and carbon, which microorganisms need to live. The final end product of mineralization is nutrients in the mineral form. Plants require nutrients to be in mineral form to take them up from soil. Therefore, all nutrients in organic matter must undergo mineralization before they can be used by living organisms. For example, consider a protein molecule containing carbon, nitrogen, phosphorus and sulfur. When microorganisms mineralize the protein molecule, it may undergo several changes to simpler organic molecules before the carbon is converted to carbon dioxide, the nitrogen to ammonium, the phosphorus to phosphate and the sulfur to sulfate. Immobilization of nutrients. Immobilization of organic matter is the opposite process to mineralization. In immobilization, mineralized nutrients are incorporated into organic molecules within living cells. The process of immobilization is very important because it relocates mineral nutrients into pools within the soil that have a relatively rapid turnover time, making them available to plants and preventing their loss by leaching. Plants are generally not efficient at competing with microorganisms for mineral nutrients in soil. C/N ratio of organic matter and why is it important? The C/N ratio of organic matter means the amount of carbon relative to the amount of nitrogen present. There is always more carbon than nitrogen in organic matter. It is usually written as C/N and as a single number because it expresses how much more carbon than nitrogen there is. For example, if the ratio is 20, this means that there are 20 grams of carbon for each gram of nitrogen in that kind of organic matter. If the ratio is 100, it means that there are 100 grams of carbon for each gram of nitrogen. So if the number is low, it means that the amount of carbon is reasonably similar to the amount of nitrogen. If the ratio is a large number, it means that there is considerably more carbon than nitrogen. The C/N ratio does not tell us what form the carbon and nitrogen are in, just how much is there. The C/N ratio is introductory agriculture. 35. Important because of what happens when organic matter is incorporated into soils. First, the larger organisms like mites and soil animals break it into smaller pieces. Then the fungi and bacteria start to decompose it. They secrete enzymes to break up the chemical compounds it is made of. When the enzymes have disrupted the compounds, the bacteria and fungi can use some of the parts released in this process as nutrients. For example, if the enzyme is degrading a protein, the microbe would be able to use the carbon, nitrogen and sulfur if there is some for its own cell wall structure and cellular contents. Excess nutrients to the requirements of the microorganisms are available for other soil organisms or plants to use. The microorganisms can access nitrogen in soil more easily than plants can, so the plants sometimes miss out. This means that if there is not enough nitrogen for all the organisms, the plants will probably be nitrogen deficient and nitrogen addition will be needed to meet the requirements of the plant. This is why incorporating organic matter into soils can change the amount of nitrogen and other nutrients available to plants. Incorporating organic matter that has a high C/N ratio will probably cause some nitrogen deficiency in the crops, plants, at least in the short term. Nitrogen cycle. The nitrogen cycle is the process by which nitrogen is converted between its various chemical forms. This transformation can be carried out through both biological and physical processes. Important processes in the nitrogen cycle include fluxation, ammonification, nitrification, and denitrification. The majority of Earth's atmosphere, 78%, is nitrogen, making it the largest pool of nitrogen. However, atmospheric nitrogen has limited availability for biological use, leading to a scarcity of usable nitrogen in many types of ecosystems. The nitrogen cycle is of particular interest to ecologists because nitrogen availability can affect the rate of key ecosystem processes, including primary production and decomposition. Human activities such as fossil fuel combustion, use of artificial nitrogen fertilizer, and release of nitrogen in wastewater have dramatically altered the global nitrogen cycle. Figure. Schematic representation of the flow of nitrogen through the environment. 36. The importance of bacteria in the cycle is immediately recognized as being a key element in the cycle, providing different forms of nitrogen compounds assimilable by high organisms. Nitrogen is present in the environment in a wide variety of chemical forms including organic nitrogen, ammonium, NH. 4 plus nitrite, no. 2 minus nitrate, no. 3 minus nitrous oxide, N. 2O nitric oxide, no, or inorganic nitrogen gas, N. 2. Organic nitrogen may be in the form of a living organism, humus, or in the intermediate products of organic matter decomposition. The processes of the nitrogen cycle transform nitrogen from one form to another. Many of those processes are carried out by microbes, either in their effort to harvest energy or to accumulate nitrogen in a form needed for their growth. The diagram above shows how these processes fit together to form the nitrogen cycle. Nitrogen fixation. Atmospheric nitrogen must be processed, or fixed, to be used by plants. Some fixation occurs in lightning strikes, but most fixations is done by free-living or symbiotic bacteria known as diazotrophs. These bacteria have the nitrogenase enzyme that combines gaseous nitrogen with hydrogen to produce ammonia, which is converted by the bacteria into other organic. Most biological nitrogen fixation occurs by the activity of monnitrogenase, found in a wide variety of bacteria and some archaea. Monnitrogenase is a complex two-component enzyme that has multiple metal-containing prosthetic groups. An example of the free-living bacteria is Azotobacter. Symbiotic nitrogen-fixing bacteria such as Rhizobium usually live in the root nodules of legumes, such as peas, alfalfa, and locust trees. Here they form a mutualistic relationship with the plant, producing ammonia in exchange for carbohydrates. Because of this relationship, legumes will often increase the nitrogen content of nitrogen-poor soils. A few non-legumes can also form such symbiosis. Today, about 30% of the total fixed nitrogen is produced industrially using the Haber-Bosch process, which uses high temperatures and pressures to convert nitrogen gas and a hydrogen source, natural gas or petroleum, into ammonia. Nitrogen assimilation. Plants take nitrogen from the soil by absorption through their roots as nitrate ions, or ammonium ions. Most nitrogen obtained by terrestrial animals can be traced back to the eating of plants at some stage of the food chain. Plants can absorb nitrate or nitrite from the soil via their root hairs. If nitrate is absorbed, it is first reduced to nitrite ions and then ammonium ions for incorporation into amino acids, nucleic acids, and chlorophyll. In plants that have a symbiotic relationship with rhizobia, some nitrogen is assimilated in the form of ammonium ions directly from the nodules. It is now known that there is a more complex cycling of amino acids between rhizobia bacteroids and plants. The plant provides amino acids to the bacteroids, so ammonia assimilation is not required, and the bacteroids pass amino acids with the newly fixed nitrogen back to the plant, thus forming an interdependent relationship. While many animals, fungi, and other introductory agriculture. 37. Heterotrophic organisms obtain nitrogen by ingestion of amino acids, nucleotides, and other small organic molecules. Other heterotrophs, including many bacteria, are able to utilize inorganic compounds such as ammonium as sole end sources. Utilization of various end sources is carefully regulated in all organisms. Ammonification. When a plant or animal dies or an animal expels waste, the initial form of nitrogen is organic. Bacteria or fungi convert the organic nitrogen within the remains back into ammonium, NH4+, a process called ammonification or mineralization. Enzymes involved are GS, GL and synthetase, cytosolic and plastic, GOGAT, glue-2-oxoglutarate aminotransferase, ferredoxin and NAD-dependent, GDH, glue dehydrogenase. Minor role in ammonium assimilation. Important in amino acid catabolism. Nitrification. The conversion of ammonia to nitrate is performed primarily by soil-living bacteria and other nitrifying bacteria. In the primary stage of nitrification, the oxidation of ammonium NH4+ is performed by bacteria such as the Nitrosomonas species, which converts ammonia to nitrites, No. 2-. Other bacterial species such as Nitrobacter are responsible for the oxidation of the nitrites into nitrates, No. 3-. It is important for the ammonia to be converted to nitrates or nitrites because ammonia gas is toxic to plants. Due to their very high solubility and because soils are largely unable to retain anions, nitrates can enter groundwater. Elevated nitrate in groundwater is a concern for drinking water use because nitrate can interfere with blood oxygen levels in infants and cause methemoglobinemia or blue baby syndrome. Where groundwater recharges stream flow, nitrate-enriched groundwater can contribute to eutrophication, a process that leads to high algal population and growth, especially blue-green algal populations. While not directly toxic to fish life, like ammonia, nitrate can have indirect effects on fish if it contributes to this eutrophication. Nitrogen has contributed to severe eutrophication problems in some water bodies. Since 2006, the application of nitrogen fertilizer has been increasingly controlled in Britain and the United States. This is occurring along the same lines as control of phosphorus fertilizer, restriction of which is normally considered essential to the recovery of eutrophied water bodies. Denitrification. Denitrification is the reduction of nitrates back into the largely inert nitrogen gas N2, completing the nitrogen cycle. This process is performed by bacterial species such as 38. Pseudomonas and Clostridium in anaerobic conditions. They use the nitrate as an electron acceptor in the place of oxygen during respiration. These facultatively anaerobic bacteria can also live in aerobic conditions. Denitrification happens in anaerobic conditions, e.g. waterlogged soils. The denitrifying bacteria use nitrates in the soil to carry out respiration and consequently produce nitrogen gas, which is inert and unavailable to plants. Anaerobic ammonia oxidation. In this biological process, nitrite and ammonia are converted directly into molecular nitrogen N2 gas. This process makes up a major proportion of nitrogen conversion in the oceans. Other processes. Though nitrogen fixation is the primary source of plant-available nitrogen in most ecosystems, in areas with nitrogen-rich bedrock, the breakdown of this rock also serves as a nitrogen source. Essential plant nutrients. 16 plant food nutrients are essential for proper crop development. Each is equally important to the plant, yet each is required in vastly different amounts. These differences have led to the grouping of these essential elements into three categories: primary, macro, nutrients, secondary nutrients, and micronutrients. Primary, macro, nutrients. Primary, macro, nutrients are nitrogen, phosphorus, and potassium. They are the most frequently required in a crop fertilization program. Also, they are needed in the greatest total quantity by plants as fertilizer. Nitrogen, necessary for formation of amino acids, the building blocks of protein. It is essential for plant cell division, vital for plant growth. Directly involved in photosynthesis, necessary component of vitamins, aids in production and use of carbohydrates and affects energy reactions in the plant. Phosphorus, involved in photosynthesis, respiration, energy storage and transfer, cell division, and enlargement. Promotes early root formation and growth, improves quality of fruits, vegetables, and grains, vital to seed formation, helps plants survive harsh winter conditions, increases water use efficiency and hastens maturity. Potassium, carbohydrate metabolism and the breakdown and translocation of starches, increases photosynthesis, increases water use efficiency, essential to protein synthesis, important in fruit formation, activates enzymes and controls their reaction rates, improves quality of seeds and fruit, improves winter hardiness and increases disease resistance. Introductory Agriculture. 39. Secondary nutrients. The secondary nutrients are calcium, magnesium, and sulfur. For most crops, these three are needed in lesser amounts than the primary nutrients. They are growing in importance in crop fertilization programs due to more stringent clean air standards and efforts to improve the environment. Calcium, utilized for continuous cell division and formation. Involved in nitrogen metabolism, reduces plant respiration, aids translocation of photosynthesis from leaves to fruiting organs, increases fruit set, essential for nut development in peanuts and stimulates microbial activity. Magnesium, key element of chlorophyll production, improves utilization and mobility of phosphorus, activator and component of many plant enzymes, directly related to grass tetany, increase iron utilization in plants and influences earliness and uniformity of maturity. Sulfur, integral part of amino acids, helps develop enzymes and vitamins, promotes nodule formation on legumes, aids in seed production and necessary in chlorophyll formation, though it isn't one of the constituents. Micronutrients. The micronutrients are boron, chlorine, copper, iron, manganese, molybdenum, and zinc. These plant food elements are used in very small amounts, but they are just as important to plant development and profitable crop production as the major nutrients. Especially, they work behind the scene as activators of many plant functions. Iron promotes formation of chlorophyll, acts as an oxygen carrier and reactions involving cell division and growth. Manganese functions as a part of certain enzyme systems, aids in chlorophyll synthesis and increases the availability of PNCA. Zinc aids plant growth hormones and enzyme system, necessary for chlorophyll production, necessary for carbohydrate formation, and necessary for starch formation and aids in seed formation. Copper catalyzes several plant processes, major function in photosynthesis, major function in reproductive stages, indirect role in chlorophyll production, increases sugar content, intensifies color and improves flavor of fruits and vegetables. Molybdenum, required to form the enzyme, nitrate reductase, which reduces nitrates to ammonium in plant, aids in the formation of legume nodules and needed to convert inorganic phosphates to organic forms in the plant. 40. Boron, essential of germination of pollen grains and growth of pollen tubes, essential for seed and cell wall formation, promotes maturity, necessary for sugar translocation and affects nitrogen and carbohydrate. Chlorine, not much information about its functions, interferes with pea uptake and enhances maturity of small grains on some soils. Plants need 16 different nutrients in order to grow, live and produce their flowers and fruits. In addition to these 13 nutrients listed above, plants require 3 more nutrients, i.e. carbon, hydrogen, and oxygen, which are extracted from air and water to make up the bulk of plant weight. They absorb the other 15 are gained through the soil. When the soil does not offer everything a plant needs, you improve it by adding fertilizer, soil conditioners and lime. Fertilizer, a synthetic chemical or natural inorganic substance, either manufactured or mined and added to soil or land to increase its fertility. Manure is organic material, mostly derived from animal feces except in the case of green manure, which can be used as organic fertilizer in agriculture. Manures contribute to the fertility of the soil by adding organic matter and nutrients, such as nitrogen, that are trapped by bacteria in the soil. Higher organisms then feed on the fungi and bacteria in a chain of life that comprises the soil food web. It is also a product obtained after decomposition of organic matter like cow dung, which replenishes the soil with essential elements and add humus to the soil. Difference between inorganic and organic fertilizer. Inorganic fertilizer are classified as those fertilizers that are synthesized or mined from non-living materials. Also known as chemical fertilizer, inorganic fertilizer are considered quick-release fertilizer, that is, the rate in which fertilizer release nutrients for the plant to absorb is relatively fast. In contrast, organic fertilizer are derived from living materials. These fertilizers are considered slow release because it takes longer for soil microbes to release the nutrients so the plant can absorb them. Complete versus balanced. Inorganic fertilizer come in single nutrient or multinutrient formulas. Multinutrient formulas include complete and balanced fertilizer, which contain basic nutrients such as nitrogen, phosphorus and potassium, as well as secondary and micronutrients such as calcium, magnesium, boron and manganese. The percentage of nitrogen, phosphorus and potassium contained in both complete and balanced fertilizer is indicated by 3 numbers on the package. For example, a 5-1-0-5 formula is a complete fertilizer containing 5% nitrogen, 10% phosphorus and 5% potassium. Balanced fertilizer are those that contain equal nutrient amounts, such as the 10th of October 10 formula. Introductory Agriculture. 41. Slow release and specially formulated. Other types of inorganic fertilizer include slow release formulas. These formulas contain larger molecules that are coated, helping them to break down slowly in the soil. A typical slow release fertilizer releases nutrients over a period of 50 days to a year, reducing the chance of burning the plant or root system. Specially formulated inorganic fertilizer are those that are created for a specific type of plant. These special formulas include plant foods for azaleas, rhododendron or roses. Specially formulated fertilizer are usually highly acidic and should be used only on the plants for which they are indicated. Nitrogen fertilizer. Inorganic nitrogen fertilizer come in many different forms, such as ammonium nitrate, potassium nitrate, calcium nitrate and urea. These fertilizer contain high levels of nitrogen, one of the most vital nutrients for plant growth. However, these inorganic fertilizer tend to increase the pH of the soil upon application, increasing the chances of burn and damage to seedlings. Others pull moisture from the air, making them difficult to apply and store. Phosphorus fertilizer. Inorganic phosphorus fertilizer such as rock phosphate remain in the soil years after the initial application. Rock phosphate works only in acidic soils, as the nutrients do not break down for plants in neutral or alkaline soils. Superphosphates are other forms of phosphorus fertilizer. These do not affect the pH of the soil upon application, while ammonium phosphates come in water-soluble, granular forms. Potassium fertilizer. Inorganic potassium fertilizer include potassium sulfate and potassium nitrate, as well as muriate of potash, also known as potassium chloride. Muriate of potash is the most commonly used potassium fertilizer. In some cases, plants may be sensitive to chloride. If a plant is sensitive to chloride, potassium sulfate, also known as sulfate of potash, is a better choice as it does not contain chloride. Potassium nitrate is easy to apply because it does not pull moisture from the air, but it does slightly increase the pH of the soil upon application. Types of organic fertilizer. Organic fertilizer come in the different forms. The most common organic fertilizer is animal manure. Fish emulsion, bone meal, cottonseed meal, blood meal and sludge are other forms of organic fertilizer. Advantages. In the case of both kinds of fertilizer, using them in the correct amounts leads to more fertile soil. Some of the specific advantages of organic fertilizer, like sludge or manure, include improved soil texture, water retention and resistance to erosion. Inorganic fertilizer. 42. However, have the potential to increase the growth rate in your plant's overall productivity more quickly. Disadvantages. While both kinds of fertilizer benefit the growth and life of your plants when used properly, they can also be highly detrimental to your plants when they are used improperly. For example, when formulas high in nitrogen are overused, they cause internal chemical burns and reduce the plant's environmental tolerances to temperature, drought and disease. In addition to causing your plants harm, excess nutrients can leach into the groundwater, causing levels of pollution to build over time. Biofertilizers. Biofertilizers are defined as preparations containing living cells or latent cells of efficient strains of microorganisms that help crop plants uptake of nutrients by their interactions in the rhizosphere when applied through seed or soil. They accelerate certain microbial processes in the soil which augment the extent of availability of nutrients in a form easily assimilated by plants. Very often microorganisms are not as efficient in natural surroundings as one would expect them to be, and therefore artificially multiplied cultures of efficient selected microorganisms play a vital role in accelerating the microbial processes in soil. Use of biofertilizers is one of the important components of integrated nutrient management, as they are cost-effective and renewable source of plant nutrients to supplement the chemical fertilizer for sustainable agriculture. Several microorganisms and their association with crop plants are being exploited in the production of biofertilizers. They can be grouped in different ways based on their nature and function. Different types of biofertilizers. Rhizobium. Rhizobium is a soil habitat bacterium, which can able to colonize the legume roots and fixes the atmospheric nitrogen symbiotically. The morphology and physiology of rhizobium will vary from free living condition to the bacteroid of nodules. They are the most efficient biofertilizer as per the quantity of nitrogen fixed concerned. They have 7 genera and highly specific to form nodule in legumes, referred as cross-inoculation group. Rhizobium inoculant was first made in USA and commercialized by private enterprise in 1930s. Initially, due to absence of efficient bradyrhizobial strains in soil, soybean inoculation at that time resulted in bumper crops, but incessant inoculation during the last 4 decades by US farmers has resulted in the buildup of a plethora of inefficient strains in soil whose replacement by efficient strains of bradyrhizobia has become an insurmountable problem. Introductory Agriculture. 43. Azotobacter. Of the several species of Azotobacter, Acrococcum happens to be the dominant inhabitant in arable soils capable of fixing N2, 2 to 15 mgN2 fixed per gram of carbon source in culture media. The bacterium produces abundant slime which helps in soil aggregation. The numbers of Acrococcum in Indian soils rarely exceeds 105 per gram soil due to lack of organic matter and the presence of antagonistic microorganisms in soil. Azospirillum. Azospirillum lipoforum and Abracillens, Spirillum lipoforum in earlier literature, are primary inhabitants of soil, the rhizosphere and intercellular spaces of root cortex of gramineous plants. They perform the associative symbiotic relation with the gramineous plants. The bacteria of genus Azospirillum are N2 fixing organisms isolated from the root and above ground parts of a variety of crop plants. They are gram-negative, Vibrio or Spirillum having abundant accumulation of polybeta-hydroxybutyrate, 70% in cytoplasm. 5 species of Azospirillum have been described to date: Abracillens, A. lipoforum, A. amazonens, A. halopreferens and A. racans. The organism proliferates under both anaerobic and aerobic conditions, but it is preferentially microaerophilic in the presence or absence of combined nitrogen in the medium. Apart from nitrogen fluxation, growth-promoting substance production, IAA, disease resistance and drought tolerance are some of the additional benefits due to Azospirillum inoculation. Cyanobacteria. Both free-living as well as symbiotic cyanobacteria, blue-green algae, have been harnessed in rice cultivation in India. A composite culture of BGA having heterocystis nostoc, Anabina, Olosira etc. is given as primary inoculum in trays, polythene line pots and later mass multiplied in the field for application as soil-based flakes to the rice-growing field at the rate of 10 kg per hectare. The final product is not free from extraneous contaminants and not very often monitored for checking the presence of desired algal flora. Once so much publicized as a biofertilizer for the rice crop, it has not presently attracted the attention of rice. 44. Growers all over India except pockets in the southern states, notably Tamil Nadu. The benefits due to algalization could be to the extent of 20 to 30 kg N per hectare under ideal conditions, but the labor-oriented methodology for the preparation of BGA biofertilizer is in itself a limitation. Quality control measures are not usually followed except perhaps for random checking for the presence of desired species qualitatively. Azolla. Azolla is a free-floating water fern that floats in water and fixes atmospheric nitrogen in association with nitrogen-fixing blue-green alga Anabina azocli. Azolla fronds consist of sporophyte with a floating rhizome and small overlapping bilobe leaves and roots. Rice-growing areas in Southeast Asia and other third world countries have recently been evincing increased interest in the use of the symbiotic N2 fixing water fern Azolla either as an alternate nitrogen sources or as a supplement to commercial nitrogen fertilizer. Azolla is used as biofertilizer for wetland rice and it is known to contribute 40 to 60 kg N per hectare per rice crop. Phosphate solubilizing microorganisms, PSM. Several soil bacteria and fungi, notably species of Phosphate solubilizing microorganisms, PSM. Several soil bacteria and fungi, notably species of Pseudomonas, Bacillus, Penicillium, Aspergillus etc. secrete organic acids and lower the pH in their vicinity to bring about dissolution of bound phosphates in soil. Increased yields of wheat and potato were demonstrated due to inoculation of peat-based cultures of Bacillus polymyxer and Pseudomonas striata. Currently, phosphate solubilizers are manufactured by agricultural universities and some private enterprises and sold to farmers through governmental agencies. These appear to be no check on either the quality of the inoculants marketed in India or the establishment of the desired organisms in the rhizosphere. AM fungi. The transfer of nutrients, mainly phosphorus and also zinc and sulfur from the soil milieu to the cells of the root cortex is mediated by intracellular obligate fungal endosymbionts of the genera Glomus, Gigaspora, Acolospora, Sclerocysts and Endogone, which possess vesicles for storage of nutrients and arbuscles for funneling these nutrients into the root system. By far, the commonest genus appears to be Glomus, which has several species distributed in soil. Availability for pure cultures of AM arbuscular mycorrhiza fungi is an impediment in large-scale production despite the fact that beneficial effects of AM fungal inoculation to plants have been repeatedly introductory agriculture. 45. Shown under experimental conditions in the laboratory, especially in conjunction with other nitrogen fixers. Silicate solubilizing bacteria, SSB. Microorganisms are capable of degrading silicates and aluminum silicates. During the metabolism of microbes, several organic acids are produced and these have a dual role in silicate weathering. They supply H+ ions to the medium and promote hydrolysis in the organic acids like citric, oxalic acid, keto acids and hydroxycarbolic acids which from complexes with cations, promote their removal and retention in the medium in a dissolved state. The studies conducted with the Bacillus sp. isolated from the soil of granite crusher yard showed that the bacterium is capable of dissolving several silicate minerals under in vitro condition. The examination of anthropogenic materials like cement, agro inputs like superphosphate and rock phosphate exhibited silicate solubilizing bacteria to a varying degree. The bacterial isolates made from different locations had varying degree of silicate solubilizing potential. Soil inoculation studies with selected isolate with red soil, clay soil, sand and hilly soil showed that the organisms multiplied in all types of soil and released more of silica and the available silica increased in soil and water. Rice responded well to application of organic siliceous residue like rice straw, rice husk and black ash at 5 t per hectare. Combining SSB with these residues further resulted in increased plant growth and grain yield. This enhancement is due to increased dissolution of silica and nutrients from the soil. Plant growth promoting rhizobacteria, PGPR. The group of bacteria that colonize roots or rhizosphere soil and beneficial to crops are referred to as plant growth promoting rhizobacteria, PGPR. The PGPR inoculants currently commercialized that seem to promote growth through at least one mechanism: suppression of plant disease, termed bioprotectants; improved nutrient acquisition, termed biofertilizers; or phytohormone production, termed biostimulants. Species of Pseudomonas and Bacillus can produce as yet not well characterized phytohormones or growth regulators that cause crops to have greater amounts of fine roots which have the effect of increasing the absorptive surface of plant roots for uptake of water and nutrients. These PGPR are referred to as biostimulants, and the phytohormones they produce include indole acetic acid, cytokinins, gibberellins, and inhibitors of ethylene production. Recent advances in molecular techniques also are encouraging in that tools are becoming available to determine the mechanism by which crop performance is improved using PGPR and track survival and activity of PGPR organisms in soil and roots. The science of PGPR is at the stage where genetically modified PGPR can be produced. PGPR with antibiotic, phytohormone and siderophore production can be made. Despite of promising results, biofertilizers has not got widespread application in agriculture mainly because of the variable response of plant species or genotypes to inoculation. 46. Depending on the bacterial strain used. Differential rhizosphere effect of crops in harboring a target strain or even the modulation of the bacterial nitrogen fixing and phosphate solubilizing capacity by specific root exudates may account for the observed differences. On the other hand, good competitive ability and high saprophytic competence are the major factors determining the success of a bacterial strain as an inoculant. Studies to know the synergistic activities and persistence of specific microbial populations in complex environments, such as the rhizosphere, should be addressed in order to obtain efficient inoculants. In this regards, research efforts are made at Agricultural College and Research Institute, Madurai to obtain appropriate formulations of microbial inoculants incorporating nitrogen fixing, phosphate and silicate solubilizing bacteria and plant growth promoting rhizobacteria which will help in promoting the use of such beneficial bacteria in sustainable agriculture. Liquid biofertilizers. Biofertilizers are such as rhizobium, azospirillum and phosphobacteria provide nitrogen and phosphorus nutrients to crop plants through nitrogen fixation and phosphorus solubilization processes. These biofertilizers could be effectively utilized for rice, pulses, millets, cotton, sugarcane, vegetable and other horticulture crops. Biofertilizers is one of the prime input in organic farming, not only enhances the crop growth and yield but also improves the soil health and sustains soil fertility. At present, biofertilizers are supplied to the farmers as carrier-based inoculants. As an alternative, liquid formulation technology has been developed in the Department of Agricultural Microbiology, TNAU, Coimbatore, which has more advantages than the carrier inoculants. Advantages. The advantages of liquid biofertilizer over conventional carrier-based biofertilizer are listed below. Longer shelf life -12-24 months. No contamination. No loss of properties due to storage up to 45 C. Greater potentials to fight with native population. Introductory agriculture. 47. High populations can be maintained more than 109 cells per milliliter up to 12 months to 24 months. Easy identification by typical fermented smell. Cost saving on carrier material, pulverization, neutralization, sterilization, packing and transport. Quality control protocols are easy and quick. Better survival on seeds and soil. No need of running biofertilizer production units throughout the year. Very much easy to use by the farmer. Dosages is 10 times less than carrier-based powder biofertilizer. High commercial revenues. High export potential. Very high enzymatic activity since contamination is nil. Characteristics of different liquid biofertilizer. Rhizobium. This belongs to bacterial group and the classical example is symbiotic nitrogen fixation. The bacteria infect the legume root and form root nodules within which they reduce molecular nitrogen to ammonia, which is reality utilized by the plant to produce valuable proteins, vitamins and other nitrogen-containing compounds. The site of symbiosis is within the root nodules. It has been estimated that 40 to 250 kg N per hectare per year is fixed by different legume crops by the microbial activities of rhizobium. Physical features of liquid rhizobium. Dull white in color. No bad smell. No foam formation. pH 6.8-7.5. Azospirillum. It belongs to bacteria and is known to fix a considerable quantity of nitrogen in the range of 20 to 40 kg N per hectare in the rhizosphere in non-leguminous plants such as cereals, millets, oilseeds, cotton etc. The efficiency of azospirillum as a biofertilizer has increased because of its ability of inducing abundant roots in several plants like rice, millets and oilseeds even in upland conditions. Considerable quantity of nitrogen fertilizer up to 25-30% can be saved by. 48. The use of azospirillum inoculant. The genus Azospirillum has 3 species viz. A. lipoforum, A. brasiliensis and A. amazonensis. These species have been commercially exploited for the use as nitrogen supplying biofertilizer. One of the characteristics of azospirillum is its ability to reduce nitrate and denitrify. Both A. lipoforum and A. brasiliensis may comprise of strains which can actively or weakly denitrify or reduce nitrate to nitrite and therefore, for inoculation preparation, it is necessary to select strains which do not possess these characteristics. Azospirillum lipoforum present in the roots of some of tropical forage grasses which is digitaria, panicum, brachiaria, maize, sorghum, wheat and rye. Physical features of liquid azospirillum. The color of the liquid may be blue or dull white. Bad odors confirms improper liquid formulation and may be concluded as mere broth. Production of yellow gummy color materials confirms the quality product. Acidic pH always confirms that there is no azospirillum bacteria in the liquid. Azospirillum cultures synthesize considerable amount of biologically active substances like vitamins, nicotinic acid, indole acetic acids, gibberellins. All these hormones, chemicals helps the plants in better germination, early emergence, better root development. Role of liquid azospirillum under field conditions. Stimulates growth and imparts green color which is a characteristic of a healthy plant. Aids utilization of potash, phosphorus and other nutrients. Encourage plumpness and succulence of fruits and increase protein percentage. Sign of non-functioning of azospirillum in the field. No growth promotion activity. Yellowish green color of leaves, which indicates no fixation of nitrogen. Azotobacter. It is the important and well-known free-living nitrogen-fixing aerobic bacterium. It is used as a biofertilizer for all non-leguminous plants, especially rice, cotton, vegetables, etc. Azotobacter cells are not present on the rhizosplane but are abundant in the rhizosphere region. The lack of organic matter in the soil is a limiting factor for the proliferation of azotobacter in the soil. Physical features of liquid azotobacter. The pigmentation that is produced by azotobacter in aged culture is melanin, which is due to oxidation of tyrosine by tyrosinase, an enzyme which has copper. The color can be noted in introductory agriculture. 49. Liquid forms. Some of the pigmentation are described below. A. crucocum produces brown-black pigmentation in liquid inoculum. A. beijerinchi produces yellow-light brown pigmentation in liquid inoculum. A. vinolandi produces green fluorescent pigmentation in liquid inoculum. A. paspali produces green fluorescent pigmentation in liquid inoculum. A. macrocytogenes produces pink pigmentation in liquid inoculum. A. incinus produces less, gumless, grayish-blue pigmentation in liquid inoculum. A. agilis produces green fluorescent pigmentation in liquid inoculum. Role of liquid azotobacter as a biocontrol agent. Azotobacter have been found to produce some antifungal substance which inhibits the growth of some soil fungi like Aspergillus, Fusarium, Curvularia, Alternaria, Helminthosporium, Fusarium etc. Acetobacter. This is a saccharophilic bacteria and associate with sugarcane, sweet potato and sweet sorghum plants and fixes 30 kgs per year. Mainly this bacterium is commercialized for sugarcane crop. It is known to increase yield by 10 to 20 t/acre and sugar content by about 10 to 15%. Effect of liquid Acetobacter diazotrophicus on sugarcane. In South India, use of Azospirillum and Phosphobacterium on the cash crop sugarcane is a regular practice for the past few years with a saving of nearly 20% of chemical nitrogen and phosphate applications. Now, it has been reported that the bacteria Acetobacter diazotrophicus which is present in the sugarcane stem, leaves, soils have a capacity to fix up to 300 kgs of nitrogen. This bacteria first reported in Brazil where the farmers cultivate sugarcane in very poor subsoil fertilized with phosphate, potassium and microelements alone, could produce yield for 3 consecutive harvests without any nitrogen fertilizer. They have recorded yield 182 to 244 tons per hectare. This leads to the assumption that active nitrogen fixing bacteria has associated within the plant. 2. Don't. Keep biofertilizer bottles away from direct heat and sunlight. Store it in cool and dry place. Don't store biofertilizer bottles under heat and sunlight. Sell only biofertilizer bottles which contain batch number, the name of the crop on which it has to be used, the date of manufacture and expiry period. Don't sell biofertilizer bottles after their expiry period is over. 50. If the expiry period is over, then discard it as it is not effective. Don't prick holes into the bottles or puncture them to pour the content. Keep biofertilizer bottles away from fertilizer or pesticide containers and they should not be mixed directly. Do not mix the biofertilizer with fungicides, insecticides, herbicides and chemical fertilizer. Liquid biofertilizer application methodology. There are 3 ways of using liquid biofertilizer. 1. Seed treatment. 2. Root dipping. 3. Soil application. Seed treatment. Seed treatment is the most common method adopted for all types of inoculants. The seed treatment is effective and economic. For small quantity of seeds, up to 5 kgs quantity, the coating can done in a plastic bag. For this purpose, a plastic bag having size 21 inches by 10 inches or big size can be used. The bag should be filled with 2 kilograms or more of seeds. The bag should be closed in such a way to trap the airs as much as possible. The bag should be squeezed for 2 minutes or more until all the seed are uniformly wetted. Then bag is opened, inflated again and shake gently. Stop shaking after each seed gets a uniform layer of culture coating. The bag is opened and the seed is dried under the shade for 20-30 minutes. For large amount of seeds, coating can be done in a bucket and an inoculant can be mixed directly with hand. Seed treatment with Rhizobium, Azotobacter, Azospirillum, along with PSM can be done. The seed treatment can be done with any of 2 or more bacteria. There is no side antagonistic effect. The important things that has to be kept in mind are that the seeds must be coated first with Rhizobium, Azotobacter or Azospirillum. When each seed get a layer of above bacteria, then PSM inoculant has to be coated as outer layer. This method will provide maximum number of each bacteria required for better results. Treatments of seed with any 2 bacteria will not provide maximum number of bacteria on individual seed. Root dipping. For application of Azospirillum, PSM on paddy transplanting, vegetable crops, this method is used. The required quantity of Azospirillum, PSM has to be mixed with 5 to 10 liters of water at one corner of the field and the roots of seedlings has to be dipped for a minimum of half an hour before transplantation. Soil application. Use 200 milliliters of PSM per acre. Mix PSM with 400 to 600 kgs of cow dung FYM along with 1/2 Introductory Agriculture. 51. Bag of rock phosphate if available. The mixture of PSM, cow dung and rock phosphate have to be kept under any tree or under shade for overnight and maintain 50% moisture. Use the mixture as soil application in rows or during leveling of soil. Azolla. Azolla is a free-floating water fern that floats in water and fixes atmospheric nitrogen in association with nitrogen-fixing blue-green alga Ranabina azocli. Azolla fronds consist of sporophyte with a floating rhizome and small overlapping bilobe leaves and roots. Rice growing areas in Southeast Asia and other third world countries have recently been evincing increased interest in the use of the symbiotic N2 fixing water fern Azolla either as an alternate nitrogen sources or as a supplement to commercial nitrogen fertilizer. Azolla is used as biofertilizer for wetland rice and it is known to contribute 40 to 60 kg NH1 per rice crop. The agronomic potential of Azolla is quite significant particularly for rice crop and it is widely used as biofertilizer for increasing rice yields. Rice crop response studies with Azolla biofertilizer in the People's Republic in China and in Vietnam have provided good evidence that Azolla incorporation into the soil as a green manure crop is one of the most effective ways of providing nitrogen source for rice. The utilization of Azolla as dual crop with wetland rice is gaining importance in Philippines, Thailand, Sri Lanka and India. The important factor in using Azolla as a biofertilizer for rice crop is its quick decomposition in soil and efficient availability of its nitrogen to rice. In tropical rice soils, the applied Azolla mineralizes rapidly and its nitrogen is available to the rice crop in very short period. The common species of Azolla are A. microphylla, A. folliculoides, A. pinnata, A. caroliniana, A. nilotica, A. rubra and A. mexicana. Method of inoculation of Azolla to rice crop. The Azolla biofertilizer may be applied in 2 ways for the wetland paddy. In the first method, fresh Azolla biomass is inoculated in the paddy field before transplanting and incorporated as green manure. This method requires huge quantity of fresh Azolla. In the other method, Azolla may be inoculated after transplanting rice and grown as dual culture with rice and incorporated subsequently. A. Azolla biomass incorporation as green manure for rice crop. Collect the fresh Azolla biomass from the Azolla nursery plot. Prepare the wetland well and maintain water just enough for easy incorporation. Apply fresh Azolla biomass 15 t/1 to the main field and incorporate the Azolla by using implements or tractor. B. Azolla inoculation as dual crop for rice. Select a transplanted rice field. 52. Collect fresh Azolla inoculum from Azolla nursery. Broadcast the fresh Azolla in the transplanted rice field on 7th day after planting, 500 kg per hectare. Maintain water level at 5 to 7.5 centimeters. Note the growth of Azolla mat 4 weeks after transplanting and incorporate the Azolla biomass by using implements or tractor or during. Maintain water level at 5 to 7.5 centimeters. Note the growth of Azolla mat 4 weeks after transplanting and incorporate the Azolla biomass by using implements or tractor or during intercultivation practices. A second bloom of Azolla will develop 8 weeks after transplanting which may be incorporated again. By the 2 incorporations, 20 to 25 tons of Azolla can be incorporated in 1 hectare rice field. Integrated nutrient management. Integrated nutrient management refers to the maintenance of soil fertility and of plant nutrient supply at an optimum level for sustaining the desired productivity through optimization of the benefits from all possible sources of organic, inorganic and biological components in an integrated manner. Integrated nutrient management concept has 3 components. 1. Regulated nutrient supply for optimum crop growth and higher productivity. 2. Improvement and maintenance of soil fertility. 3. Zero adverse impact on ecosystem quality by balanced fertilization of organic manures, inorganic fertilizer and bioinoculant. Integrated nutrient management determinants are 1. Nutrient requirement of cropping system as a whole. 2. Soil fertility status and special management needs to overcome soil problems, if any. 3. Local availability of nutrients resources, organic, inorganic and biological sources. 4. Economic conditions of farmers and profitability of proposed INM option. 5. Social acceptability. 6. Ecological considerations. 7. Impact on the environment. Advantages of integrated nutrient management. 1. Enhances the availability of applied as well as native soil nutrients. 2. Synchronizes the nutrient demand of the crop with nutrient supply from native and introductory agriculture. 53. Applied sources. 3. Provides balanced nutrition to crops and minimizes the antagonistic effects resulting from hidden deficiencies and nutrient imbalance. 4. Improves and sustains the physical, chemical and biological functioning of soil. 5. Minimizes the deterioration of soil, water and ecosystem by promoting carbon sequestration, reducing nutrient losses to ground and surface water bodies and to atmosphere. Integrated nutrient management components are soil source, mobilizing unavailable nutrients into use appropriate crop varieties, cultural practices and cropping system. Mineral fertilizer, super granules, coated urea, direct use of locally available rock PO4 in acid soils, single superphosphate, SSP, MOP and micronutrient fertilizer. Organic sources, byproducts of farming and allied industries. FYM, droppings, crop waste, residues, sewage, sludge, industrial waste. Biological sources, use of biofertilizer i.e. microbial inoculant substitute 15 to 40 kg N per hectare. Classification of crops. Importance of classifying the crop plants. 1. To get acquainted with crops. 2. To understand the requirement of soil and water different crops. 3. To know adaptability of crops. 4. To know the growing habit of crops. 5. To understand climatic requirement of different crops. 6. To know the economic produce of the crop plant and its use. 7. To know the growing season of the crop. 8. Overall to know the actual condition required to the cultivation of plant. Classification based on climate. Tropical. Crops grow well in warm and hot climate, e.g. rice, sugarcane, jowar etc. Temperate. Crops grow well in cool climate, e.g. wheat, oats, gram, potato etc. Classification based on growing season. 1. Kharif, rainy, monsoon crops, the crops grown in monsoon months from June to OCT. 54. November, require warm, wet weather at major period of crop growth, also required short day length for flowering, e.g. cotton, rice, jowar, bajara. 2. Rabi, winter, cold seasons crops, require winter season to grow well from OCT to March month. Crops grow well in cold and dry weather. Require longer day length for flowering, e.g. wheat, gram, sunflower etc. 3. Summer, Z crops, crops grown in summer month from March to June. Require warm day weather for major growth period and longer eye length for flowering, e.g. groundnuts, watermelon, pumpkins, gourds. Use agronomic classification. 1. Grain crops, may be cereals as millet. Cereals are the cultivated grasses grown for their edible starchy grains. The larger grain used as staple food is cereals, e.g. rice, jowar, wheat, maize, barley, and millets are the small grain cereals which are of minor importance as food, e.g. bajara. 2. Pulse, legume crops, seeds of leguminous crops plant used as food. On splitting they produce dal which is rich in protein, e.g. green gram, black gram, soybean, pea, cowpea etc. 3. Oilseeds crops, crop seeds are rich in fatty acids, are used to extract vegetable oil to meet various requirements, e.g. groundnut, mustard, sunflower, sesame, linseed etc. 4. Forage crop, it refers to vegetative matter fresh as preserved utilized as food for animals. Crop cultivated and used for fikla, hay, silage, sorghum, elephant grass, guinea grass, berseem and other pulse bajara etc. 5. Fiber crops, ground for fiber yield. Fiber may be obtained from seed, e.g. cotton, steam, jute, mesta, sun hemp, flax. 6. Roots crops, roots are the economic produce in root crop, e.g. sweet, potato, sugar beet, carrot, turnip etc. 7. Tuber crop, crop whose edible portion is not a root but a short thickened underground stem, e.g. potato, elephant, yam. 8. Sugar crops, the two important crops are sugarcane and sugar beet cultivated for production for sugar. 9. Starch crops, grown for the production of starch, e.g. tapioca, potato, sweet potato. 10. Dreg crop, used for preparation for medicines, e.g. tobacco, mint, pyrethrum. 11. Spices and condiments, spices crops, crop plants as their products are used to flavor introductory agriculture. 55. Taste and sometime color the fresh preserved food, e.g. ginger, garlic, chili, cumin, onion, coriander, cardamom, pepper, turmeric etc. 12. Vegetables crops, may be leafy as fruity vegetables, e.g. palak, mentha, brinjal, tomato. 13. Green manure crop, grown and incorporated into soil to increase fertility of soil, e.g. sun hemp. 14. Medicinal and aromatic crops, medicinal plants include cinchona, isabgol, opium poppy, senna, belladonna, rauwolfra, acarice and aromatic plants such as lemongrass, citronella grass, palmrosa, Japanese mint, peppermint, rose, jasmine, henna etc. Classification based on life of crops, duration of crops. 1. Seasonal crops, a crop completes its life cycle in one season karen, rabi, summer. E.g. rice, jowar, wheat etc. 2. Two seasonal crops, crops complete its life in 2 seasons, e.g. cotton, turmeric, ginger. 3. Annual crops, crops require 1 full year to complete its life in cycle, e.g. sugarcane. 4. Biennial crops, which grows in 1 year and flowers, fructifies and perishes the next year, e.g. banana, papaya. 5. Perennial crops, crops live for several years, e.g. fruit crops, mango, guava etc. Classification based on cultural method. Water. 1. Rain-fed, crops grow only on rainwater, e.g. jowar, bajara, mung etc. 2. Irrigated crops, crops grows with the help of irrigation water, e.g. chili, sugarcane, banana, papaya etc. Classification based on root system. 1. Tap root system, the main root goes deep into the soil, e.g. 2. Grape, cotton etc. 2. Adventitious, fiber rooted, the crops whose roots are fibrous shallow and spreading into the soil, e.g. cereal crops, wheat, rice etc. Classification based on economic importance. 1. Cash crop, grown for earning money, e.g. sugarcane, cotton. 56. 2. Food crops, grown for raising food grain for the population and fodder for cattle, e.g. jowar, wheat, rice etc. Classification based on know of cotyledons. 1. Monocots or monocotyledons, having 1 cotyledon in the seed, e.g. all cereals and millets. 2. Dicots or dicotyledonous, crops having two cotyledons in the seed, e.g. all legumes and pulses. Classification based on photosynthesis, reduction of CO2, dark reaction, 1. C3 plants, photorespiration is high in these plants. C3 plants have lower water use efficiency. The initial product of C assimilation in the 3C compounds. The enzyme involved in the primary carboxylation is ribulose 1, biophosphate carboxylose, e.g. rice, soybeans, wheat, barley, cottons, potato. 2. C4 plants, the primary product of C fixation is 4 carbon compounds which may be malic acid or acerbic acid. The enzymes responsible for carboxylation are phosphenol pyruvic acid carboxylose which has high affinity for CO. 2. Incapable of assimilation CO. 2. Event at lower concentration, photorespiration is negligible. Photosynthetic rates are higher in C4 than C3 plants for the same amount of stomatal opening. These are said to be drought resistant and they are able to grow better even under moisture stress. C4 plants translate photosynthesis rapidly, e.g. sorghum, maize, naptor grass, sesame etc. 3. Cam plants, casulacin acid metabolism plants, the stomata open at night and large amount of CO. 2 is fixed as a malic acid which is stored in vacuoles. During day stomata are closed. There is no possibility of CO. 2. Entry. CO. 2 which is stored as malic acid is broken down and released as CO. 2. In these plants there is negligible transpiration. C4 and cam plant have high water use efficiency. These are highly drought resistant, e.g. pineapple, sisal and agave. Classification based on length of photoperiod required for floral initiation. Most plants are influenced by relative length of the day and night, especially for floral initiation. The effect on plant is known as photoperiodism depending on the length of photoperiod required for floral ignition. Plants are classified as 1. Short day plants. Flower initiation takes place when days are short less than 10 hours, e.g. rice, jowar, green gram, black gram etc. 2. Long days plants. Require long days are more than 10 hours for floral ignition, e.g. wheat, barley. 3. Day neutral plants. Photoperiod does not have much influence for phase change for introductory agriculture. 57. These plants, e.g. cotton, sunflower. The rate of the flowering initiation depends on how short or long is photoperiod. Shorter the days, more rapid initiation of flowering in short days plants. Longer the days, more rapid are the initiation of flowering in long days plants. Classification of horticultural crops based on use of horticultural plants. Use of horticultural plants. I. Edible plants. A. Vegetables. B. Fruits. E. Ornamental plants. I. Edible plants are vegetable. A. Vegetables grown for aerial portion. 1. Cole crops, cabbage, cauliflower. 2. Legume crops, peas and beans. 3. Solanaceous crops, tomato, brinjal. 4. Cucurbits, cucumber, red pumpkin. 5. Leafy vegetables, spinach, methi. 6. Salad vegetables, lettuce, broccoli. 7. Corn vegetables, sweet corn and popcorn. B. Vegetable grown for underground portion. 1. Root crops, beet, carrot. 2. Tuber crops, yam, potato. 3. Bulb crops, onion and garlic. B. Fruits. A. Temperate, deciduous fruits. 1. Small fruits, grape, strawberry. 2. Tree fruits, apple, pear, cherry. 3. Nuts, peach, walnut. 58. B. Tropical and subtropical. 1. Herbaceous perennials, pineapple, banana. 2. Tree fruits, mango, papaya. 3. Nuts, cashew nut, arecanut. 2. Ornamental plants. 1. Flowering trees, gulmohar, neelmobor, cashew. 2. Roadside trees, neem, banyan tree, rain tree. 3. Shade-giving trees, rain tree, mahogany. 4. Flowering shrubs, nerium, hibiscus, tugger. 5. Foliage shrubs, tuja, casuarina. 6. Climbers and creepers, petrea, bignonia, ipomi. 7. Bulbous plants, canna, caladium, tuberose. 8. Hedge and edges, duranta, clerodendron, ageratum. 9. Annuals, petunia, aenia. 10. Perennials, chrysanthemum, roses. Deciduous tree, fig, guava, apple, karvanda, bay, sweet cherry, pomegranate, grape, mulberry, falsa, almond. Evergreen trees. Arecanut, dates, coconut, pineapple, banana, jackfruit, avocados, sweet orange, mandarin orange, key lime, mango, chiku, papaya, passion fruit, cashew nut. Horticultural classifications. With hundreds of thousands of plants used by mankind, it is impossible to talk about each one individually. Plants are grouped by various common characteristics to help us communicate similar ecological adaptations and cultural requirements. For example, the Introductory Agriculture. 59. Term, shade plants, indicates plants tolerant to various levels of shade. Xeric groups those plants requiring less supplemental irrigation in our climate. It is important to point out that any classification system will have plants that don't exactly fit the groupings. The following are examples of some common classifications used in horticulture. Classification by use. I. Edibles. A. Fruits. 1. Tree fruits. 2. Small fruits. B. Vegetables. 1. Warm season vegetables. 2. Cool season vegetables. C. Herbs. 1. Culinary. 2. Medicinal. D. Nuts. 2. Ornamentals. Landscape plants. A. Woody plants. 1. Trees. 2. Shrubs. 3. Vines and ground covers. B. Herbaceous plants. 1. Flowers. 2. Vines and ground covers. C. Grass, turf. 3. Potted plants, house plants, gift plants. A. Flowering gift plants. B. Foliage plants. 60. System of farming. A system is defined as a set of components that are interrelated and interact among themselves. A cropping system refers to a set of crop systems, making up the cropping activities of a farm system. Cropping system comprises all components required for the production of a particular crop and the interrelationships between them and environment. In other words, a cropping system usually refers to a combination of crops in time and space. Combination in time occurs when crops occupy different growing period and combinations in space occur when crops are interplanted. When annual crops are considered, a cropping system usually means the combination of crops within a given year. Cropping pattern. The yearly sequence and spatial arrangement of crops or of crops and fallow on a given area. Cropping system. The cropping patterns used on a farm and their interaction with farm resources, other farm enterprises, and available technology which determine their makeup. Intensive cropping. Principles. The turnaround period between one crop and another is minimized through modified land preparation. It is possible when the resources are available in plenty. X. Garden land cultivation. Cropping intensity is higher in intensive cropping system. Crop intensification technique includes intercropping, relay cropping, sequential cropping, ratoon cropping, etc. All such systems come under the general term multiple cropping. Need for intensive cropping. Cropping systems has to be evolved based on climate, soil and water availability for efficient use of available natural resources. The increase in population has put pressure on land to increase productivity per unit area, unit time and for unit resource used. This cropping system should provide enough food for the family, fodder for cattle, and generate sufficient cash income for domestic and cultivation expenses. Intensive cropping, growing number of crops on the same piece of land during the given period of time. Cropping intensity, number of crops cultivated in a piece of land per annum is cropping intensity. In Punjab and Tamil Nadu, the cropping intensity is more than 100%, i.e. around 140 to 150%. In Rajasthan, the cropping intensity is less. Introductory Agriculture. 61. Multiple cropping, the intensification of cropping in time and space dimensions. Growing two or more crops on the same field in a year. Forms of multiple cropping. Intercropping, growing two or more crops simultaneously on the same field. Crop intensification is in both time and space dimensions. There is intercrop competition during all or part of crop growth. Mixed intercropping, growing two or more crops simultaneously with no distinct row arrangement. Also referred to as mixed cropping. X, sorghum, pearl millet and cowpea are mixed and broadcasted in rain-fed conditions. B, row intercropping, growing two or more crops simultaneously where one or more crops are planted in rows. Often simply referred to as intercropping. Maize plus green gram, 1, 1, maize plus black gram, 1, 1, groundnut plus red gram, 6, 1. C. Strip intercropping, growing two or more crops simultaneously in strips wide enough to permit independent cultivation but narrow enough for the crops to interact agronomically. X, groundnut plus red gram, 6, 4, strip. D. Relay intercropping, growing two or more crops simultaneously during the part of the life cycle of each. A second crop is planted after the first crop has reached its reproductive stage of growth, but before it is ready for harvest. Often simply referred to as relay cropping. Rice fallow pulse. Advantages of intercropping. Better use of growth resources including light, nutrients and water. Suppression of weeds. Yield stability. Even if one crop fails due to unforeseen situations, another crop will yield and gives income. Successful intercropping gives higher equivalent yields, yield of base crop plus yield of intercrop, higher cropping intensity. Reduced pest and disease incidences. Improvement of soil health and agro-ecosystem. Sequential cropping, growing two or more crops in sequence on the same field in a farming year. The succeeding crop is planted after the preceding crop has been harvested. Crop intensification is only in time dimension. There is no intercrop competition. 62. Double, triple and quadruple cropping, growing 2, 3 and 4 crops, respectively, on the same land in a year in sequence. X. Double cropping, rice, cotton, triple cropping, rice, pulses, quadruple cropping, tomato, ridge gourd, amaranthus greens, baby corn, B, ratoon cropping, the cultivation of crop regrowth after harvest, although not necessarily for grain. X. Sugarcane, ratoon, sorghum, ratoon, for fodder. The various terms defined above bring out essentially two underlying principles, that of growing crops simultaneously in mixture, i.e., intercropping, and of growing individual crops in sequence, i.e., sequential cropping. The cropping system for a region or farm may comprise either or both of these two principles. Sustainable agriculture. Definition. A farming systems that are capable of maintaining their productivity and usefulness to society indefinitely and must be resource conserving, socially supportive, commercially competitive, and environmentally sound. Satisfy human food and fiber needs. Enhance environmental quality in the natural resource based upon which the agricultural economy depends. Make the most efficient use of non-renewable resources and on-farm resources and integrate, where appropriate, natural biological cycles and controls. Sustain the economic viability of farm operations. Enhance the quality of life for farmers and society as a whole. Advantages of sustainable agriculture. Production cost is low. Overall risk of the farmer is reduced. Pollution of water is avoided. Very little or no pesticide residue is ensured. Ensures both short and long-term profitability. Introductory agriculture. Introductory agriculture. 63. Disadvantages of sustainable agriculture. Since sustainable agriculture uses least quantum of inputs, naturally the output yield may also be less. Major components of sustainable agricultural system. Soil and water conservation to prevent degradation of soil productivity. Efficient use of limited irrigation water without leading to problems of soil salinity, alkalinity and high groundwater table. Crop rotations that mitigate weed, disease and insect problems, increase soil productivity and minimize soil erosion. Integrated nutrient management that reduces the need for chemical fertilizer, improves the soil health and minimize environmental pollution by conjunctive use of organics, inorganics and biofertilizer. Integrated pest management that reduces the need for agrochemicals by crop rotation, weather monitoring, use of resistant cultivar, planting time and biological pest control. Management system to control weed by preventive measures, tillage, timely intercultivation and crop rotation to improve plant health. Integrated farming system, IFS. Integration of two or more appropriate combination of enterprises like crop, dairy, piggery, fishery, poultry, beekeeping etc. for each farm according to the availability of resources to sustain and satisfy the necessities of the farmer. Definition. A farming system is a collection of distinct functional units such as crop, livestock, processing, investments and marketing activities which interact because of the joint use of inputs they receive from the environment which have the common objective of satisfying the farmers, decision makers' aims. The definition of the borders of the options depends on circumstances. Often it includes not only the farm, economic enterprise, but also the household system. Possible enterprises. Wetland-based farming system. Crop plus fish plus poultry, pigeon. Crop plus fish plus mushroom. 64. Gardenland-based farming system. Crop plus dairy plus biogas. Crop plus dairy plus biogas plus sericulture. Crop plus dairy plus biogas plus mushroom plus silviculture. Dry land-based farming system. Crop plus goat plus agroforestry. Crop plus goat plus agroforestry plus horticulture. Advantages of integrated farming system. Higher productivity, profitability, sustainability, balanced food, recycling reduces pollution, money round the year, employment generation, increase input efficiency, standard of living of the farmer increased, better utilization of land, labor, time and resources. Relay cropping. Relay cropping is essentially a special version of double cropping, where the second crop is planted into the first crop before harvest, rather than waiting until after harvest as in true double cropping. In this way, both crops share a portion of the growing season, increasing solar radiation and heat available to each. System pros and cons. Relay cropping has advantages and disadvantages compared to standard cropping practices. Advantages of relay cropping. The potential to reduce nitrate leaching, wheat acts as a scavenger crop. Increase carbon sequestration, and increase income for producers. Introductory agriculture. 65. Disadvantages relay cropping. The soybean planting process will likely stress the wheat crop and reduce yield from what would be expected of a non-disturbed crop. Likewise, wheat harvest may stress the soybean crop. The hope is that the two crops will result in greater income and profit than either single crop grown without disturbance. Relay cropping requires a greater level of management. Wheat must be planted during the soybean and corn harvest season, and planting soybean into a standing crop is a new process to most farmers. Also, pest management and control practices must account for more crops being grown in close sequence. The system may not allow time for herbicide carry-over levels to decline and may increase the potential for insect and disease infestations if these pests have more than one host in the crop sequence. Crop rotation. Crop rotation. It refers to recurrent succession of crops on the same piece of land either in a year or over a longer period of time. It is a process of growing different crops in succession on a piece of land in a specific period of time, with an objective to get maximum profit from least investment without impairing the soil fertility. Characteristics of crop rotation or principles of crop rotation. 1. It should be adaptable to the existing soil, climatic and economic factors. 2. The sequence of cropping adopted for any specific area should be based on proper land utilization. It should be so arranged in relation to the fields on the farm that the yields can be maintained and soil losses through erosion reduced to the minimum. 3. The rotation should contain a sufficient acreage of soil improving crops to maintain and also build up the own content of the soil. 4. In areas where legumes can be successfully grown, the rotation should provide for a sufficient acreage of legumes to maintain the end supply of the soil. 5. The rotation should provide roughage and pasturage for the livestock kept on farm. 6. It should be so arranged as to help in the control of weeds, plant disease and insect pests. 7. It should provide for the acreage of the most profitable cash crops adapted to the area. 66. 8. The rotation should be arranged as to make for economy in production and labor utilization exhaustive potato, sugarcane, followed by less exhaustive crops, oilseeds and pulses. 9. The crops with tap roots should be followed by those which have fibrous root system. This helps in proper and uniform use of nutrients from the soil and roots do not compete with each other for uptake of nutrients. 10. The selection of crops should be problem and need, demand base. I. According to need of people of the area and family. E. On slop lands, alternate cropping of erosion promoting and erosion resisting crops should be adopted. E. Under dryland or limited irrigation, drought tolerant crops, jowar, bajra, in low-lying and flood-prone areas, water stagnation tolerant crops, paddy, jute should be adopted. IV. Crops should suit to the farmer's financial conditions, soil and climatic conditions. 11. The crops of the same family should not be grown in succession because they act like alternate hosts for insect pests and disease pathogens and weeds associated with crops. 12. An ideal crop rotations is one which provide maximum employment to the family and farm labor. The machines and equipments are efficiently used so all the agri operations are done timely. Advantages of crop rotation. An ideal crop rotation has the following advantages. 1. There is an overall increase in the yield of crops due to maintenance of proper physical condition of the soil and its own content. 2. Inclusion of crops having different feeding zones and different nutrient requirements help in maintaining a better balance of nutrients in the soil. 3. Diversification of crops reduces the risk of financial loss from unfavorable weather conditions and damage due to pests and diseases. 4. It facilitates more even distribution of labor. 5. There is regular flow of income over the year. 6. The incidence of weeds, pests and diseases is reduced and can be kept under control. 7. Proper choice of crops in rotation helps to prevent soil erosion. 8. It supplies various needs of farmer and his cattle. Introductory agriculture. 67. 9. Agricultural operations can be done timely for all the crops because of less competition. The supervisory work also becomes easier. 10. Proper utilization of all the resources and inputs could be made by following crop rotation. Cropping systems and crop rotations followed in Maharashtra and Marathwada. Maharashtra. 1. Cotton, jowar, bajara, cotton, jowar, groundnut. 2. Sugarcane, rice, gram. 3. Cotton, groundnut, cotton, jowar, bajara, groundnut. 4. San hemp, sugarcane. 5. Six. Rice, gram. 7. Groundnut, cotton, jowar. Marathwada. 1. Mung, jowar, cotton plus 2. 2. Sunflower, jowar. 3. Soybean, jowar, safflower, gram. 4. High, jowar, gram, sunflower, safflower. 5. Bajara, gram, mung, soybean, jowar, safflower. Irrigated. 1. Cotton, groundnut, san hemp, sugarcane, groundnut. 2. Rice, gram, sunflower. 3. High, jowar, wheat, jowar, gram. 4. Jowar, sunflower, groundnut. 5. Sunflower, potato, groundnut. 6. Groundnut, wheat, vegetables. 7. Sorghum, wheat, green gram, cotton, groundnut. 8. Bajara, cabbage, groundnut, cotton, groundnut. 68. Water management. One of the major ways in which land can be improved for farming is by water management. By regulating the amount of water in the soil, aeration can be improved, activity by useful bacteria can be stimulated and crop yields can be improved. In addition, by draining or irrigating land, areas which are marginal or useless in their natural state, such as deserts or swamps, can be brought into agricultural production. It should be emphasized that drainage and irrigation are interdependent. Where irrigation is used, it is important also to provide drainage facilities, so that the irrigation water can be kept moving and not become stagnant. Similarly, in drained areas, irrigation must be applied to prevent unwanted seawater from seeping into the drained land. In other words, a balance must be carefully maintained. Irrigation. When a region does not have sufficient natural precipitation to meet the plant's moisture requirements, an artificial supply of water is necessary. This is known as irrigation. The amount of extra water needed depends much on the type of crops grown, the prevailing temperature and humidity, the kind of soil and the physical conditions of the surrounding districts. Irrigation is one of the oldest agriculture techniques practiced by men, and has many advantages over simple reliance on natural water supplies. The supply of water by irrigation is regular and reliable, whereas rainfall is often seasonal or unpredictable. In desert areas, the use of irrigation allows cultivation to take place where it would not otherwise be possible. Irrigation water supplied by rivers in flood often carries much silt and clay, which adds to the soil of the fields, enhancing fertility and thus crop yields. There are different water resources for irrigation, e.g. tube well, canal, river, lakes, etc. Drainage. Drainage is the natural or artificial removal of surface and subsurface water from an area. Many agricultural soils need drainage to improve production or to manage water supplies. In moist climates, soils may be adequate for cropping with the exception that they become waterlogged for brief periods each year, from snowmelt or from heavy rains. Soils that are predominantly clay will pass water very slowly downward, meanwhile plant roots suffocate because the excessive water around the roots eliminates air movement through the soil. Other soils may have an impervious layer of mineralized soil, called a harpen, or relatively impervious rock layers may underlie shallow soils. Drainage is especially important in tree fruit production. Soils that are otherwise excellent may be waterlogged for a week of the year, which is sufficient to kill fruit trees and cause the productivity of the land until replacements can be established. In each of these cases, appropriate drainage carries off temporary flushes of water to prevent damage to annual or perennial crops. Drier areas are often farmed by irrigation, and one would not consider drainage necessary. However, irrigation water always contains minerals and salts, which can be concentrated to introductory agriculture. 69. Toxic levels by evapotranspiration. Irrigated land may need periodic flushes with excessive irrigation water and drainage to control soil salinity. Definition of micro-irrigation. It can be defined as the application of water at low volume and frequent interval under low pressure to plant root zone. Concept of micro-irrigation. Besides the land, water also an important factor in the progress of agriculture. In vast country like India with a geographical area of 328 million hectares, less than 45% area is cultivated. Of this cultivated area, only 35%, i.e. 65 million hectares gets irrigation. Since water is the limiting factor today, we must utilize it properly and maximum benefit can take as possible, as India is second largest country in world according to population, 1202 million. The expansion of area under irrigation is essential for obtaining increased agriculture production required to feed India's growing population. The expansion could be done only by additional development, conservation and efficient management of the available water resources, i.e. use of micro-irrigation means application of optimum water according to plant requirement. This could be achieved by introducing advanced and sophisticated methods of irrigation, viz. Drip irrigation, sprinkler, etc. Drip irrigation. It is termed as trickle irrigation also and can be defined as slow and frequent application of water to the plant root zone under low pressure. The drip irrigation is an advanced method of irrigation suitable for water scarcity areas. It uses the water very economically and efficient. It applies the correct quantity of water to the plant at the root zone through a network of tubings. The design of drip system is simple. These requirements are pipes, accessories and other equipment. This method can be very well adopted for wide-spaced crops like coconut, mango, sapota, banana, etc. A drip irrigation system consists essentially of mainline, sub-mains, lateral, drippers, filters and other small fittings and accessories like valves, pressure regulators, pressure gauge, fertilizer application components, etc. Advantages of drip irrigation. Maximum use of available water. No water being available to weeds. Maximum crop yield. High efficiency in the use of fertilizer. Less weed growth and restricts population of potential hosts. 70. Low labor and relatively low operation cost. No soil erosion. Improved infiltration in soil of low intake. Ready adjustment to sophisticated automatic control. No runoff of fertilizer into groundwater. Less evaporation losses of water as compared to surface irrigation. Improved seed germination. Decreased tillage operations. Disadvantages of drip irrigation. In spite of the fact that drip irrigation has so many potential benefits, there are certain limitations also. There are as follow. Sensitivity to clogging. Moisture distribution problem. Salinity hazards. High cost compared to furrow. High skill is required for design, install and operation. Sprinkler irrigation. In the sprinkler method of irrigation, water is sprayed into the air and allowed to fall on the ground surface somewhat resembling rainfall. The spray is developed by the flow of water under pressure through small orifices or nozzles. The pressure is usually obtained by pumping. With careful selection of nozzle sizes, operating pressure and sprinkler spacing, the amount of irrigation water required to refill the crop root zone can be applied nearly uniform at the rate to suit the infiltration rate of soil. Advantages of sprinkler irrigation. Elimination of the channels for conveyance, therefore no conveyance loss. Suitable to all types of soil except heavy clay. Suitable for irrigating crops where the plant population per unit area is very high. It is most suitable for oilseeds and other cereal and vegetable crops. Water saving, introductory agriculture. 71. Closer control of water application, convenient for giving light and frequent irrigation and higher water application efficiency. Increase in yield. Mobility of system. May also be used for undulating area. Saves land as no bunds etc. are required. Influences greater conducive microclimate. Areas located at a higher elevation than the source can be irrigated. Possibility of using soluble fertilizer and chemicals. Less problem of clogging of sprinkler nozzles due to sediment-laden water. Disadvantages of sprinkler irrigation. Incurs high operation expenses due to the energy needed for pumping, labor and relatively large investment in equipment. Sensitivity to wind, causing evaporation losses. The unavoidable wetting of foliage in field crops results in increased sensitivity to diseases. Debris and sediments can cause clogging. Capital cost is high with greater operational costs due to high energy requirements. Soil-plant, water, relationships. Soil-plant-water relation relates to the physical properties of soil and plants that affect the movement, retention and use of water. These relations must be considered in designing and operation systems. Soil is a storehouse of plant nutrients, a habitat for bacteria, an anchorage for plant and a reservoir that hold the water needed for plant growth. The amount of water a soil can hold in available for plant use is determined by its physical properties. This amount determines the length of time a plant can survive without water being added. It determines both the frequency of irrigation and the capacity of irrigation system needed to ensure continuous crop growth. Soil is a three-phase system comprising of the soil phase made of mineral and organic matter and various chemical compounds, the liquid phase called soil moisture and the gaseous phase called the soil air. They also contain variety of living and organism. Two important physical properties of soil, which differ the supply of water and air in soil, are texture and structure. 72. Classification of soil water. Although the soil water cannot be demarcated, yet for the sake of understanding, and as per utility of that water to plants, it is mainly classified into three categories. A. Hygroscopic water. B. Capillary water. C. Gravitational water. A. Hygroscopic water. It is the part of soil water which is very tightly held on the surface of soil particle in very thin film by the forces of adhesion and cohesion. It is mostly in vapor from and the force with which it is held on the surface of soil particle is estimated at about 10,000 atmosphere towards the inner side and about 31 atmosphere at the outer side. Hygroscopic water is not available to the plant and agriculturally it has no importance. B. Capillary water. The capillary water is that water which held in the soil in excess of hygroscopic water, but is up to the point where the gravity pull begins to move the water downwards when free drainage conditions exist in the soil. Capillary water is rather loosely held water, from 31 atmospheres to 1/3 of an atmosphere tension, and it moves more slowly than free water. It can move in any direction, but always in the direction greatest tension. A soil, which has a finer texture and granular indicating larger proportion of micropores than macropores, holds more amount of capillary water than coarse textured single grain sandy soil having more percentage of macropores. Similarly, soils rich in organic matter content also hold a much greater quantity of capillary water. C. Gravitational water. When all the pores, large and small, are filled, the soil is to be saturated, that is at its maximum retentive capacity. Within 2 to 3 days after heavy irrigation, the gravitational water or free water gets drained away and the moisture content at this stage in the surface soil is said to be at field capacity. The moisture content at this stage is about 1/3 atmosphere. Permanent wilting point (PWP) or wilting point (WP) is defined as the minimal point of soil moisture the plant requires not to wilt and water present in the soil at 31 atmosphere. If moisture decreases to this or any lower point, a plant wilts and can no longer recover its turgidity when placed in a saturated atmosphere for 12 hours. Moisture present between field capacity to permanent wilting point (PWP) is called as available water. This amount of water is biologically introductory agriculture. 73. Available to the plant for their physiological requirement. There have been various methods developed to determine the water requirement for specific plants. Critical growth stages and water requirements of important crops are presented in the table. Table. Critical growth stages and water requirements of important crops. Crop critical growth stages average crop duration, days, water requirement Rice tiller initiation, flowering and milky stage 90, 900, 250. Crop critical growth stages average crop duration, days, water requirement Wheat crown root initiation, flowering, joining, milky and tilling 135 400 to 450. Crop critical growth stages average crop duration, days, water requirement Pulses flower initiation and pod filling 90 to 120 250 to 300. Crop critical growth stages average crop duration, days, water requirement Groundnut pegging and pod formation 105 600 to 450. Crop critical growth stages average crop duration, days, water requirement Sugarcane emergence, tiller formation and elongation 330, 1400, 3000. Crop critical growth stages average crop duration, days, water requirement Banana early vegetative phase, bunch initiation and flowering 300, 3000. Crop critical growth stages average crop duration, days, water requirement Cassava rooting, early tuberization and tuber development 300, 400 to 750. 74. Location of soil moisture measurement and moisture measuring instruments. Location of soil moisture measurement. Soil moisture measurement should be made in that part of soil from which plant roots extracts their moisture and according to the moisture extraction pattern of the crop. One measurement should be made in upper quarter of the root zone and one or more two measurement at lower levels. Moisture measuring instruments. Various instruments for measuring are available commercially. These instruments may not give an accurate results as the soil sampling and drying procedure but are sufficiently accurate for practical purpose. The types of instruments commonly in use to measure the soil moisture are 1. Tensiometer 2. Electrical resistance measuring devices 3. Neutron moisture meter. Soil and water conservation methods. Management practices are strip cropping. This consists of growing erosion permitting crops and erosion resisting crops in alternate strips. The erosion permitting crops are cotton, jawar, bajara, etc., which allow the runoff water to flow freely within the rows. The erosions resisting crops are mostly legumes like groundnut, moth bean, Phaseolus aconituleus, Culati, Dolichos biflorus, soybean, Glycine max, which spread and cover the soil and do not allow runoff water to carry much soil with it. The soil which flows from the strips growing erosion permuming crops is caught by the alternating springs. In selecting a suitable legume crop, it should be seen that the maximum canopy and root development of the crop coincide with the period of high intensity of rainfall. B. Mulching. A mulch is natural or artificially applied layer of plant residues or other material on the surface of the soil with the object of moisture conservation, temperature control, prevention of surface compaction or crust formation, reduction of runoff and erosion, improvement in soil structure and weed control. Artificial mulches of different kinds such as jawar or bajara stubbles, paddy straw or husk, sawdust etc. increase absorption of water and minimize evaporation. They also control runoff and soil losses. C. Rotation of crops. Rotation means growing a set of crops in a regular succession over the same field within a specified period of time. Continuous growing of jawar or bajara crop causes more erosion, but if followed by a legume crop viz, halga, matki or gram which covers the soil causes less erosion. Rotation also helps in removal. Introductory agriculture. 75. Of plant nutrients in a uniform way from future depth of soil and in maintaining the fertility of the soil in dry farming region of Maharashtra. Adoption of gram jawar rotation not only helps in conservation of moisture but also in increasing the crop yields. The beneficial effect of rotation can be seen from the following table. D. Contour cultivation, tillage operations viz, plowing, harrowing, sowing and interculture should be done across the slope of land. This will help in creating obstructions to the flow of water at every furrow, which acts like a small bund and results in uniform distribution of water. This helps more nitration of water, less runoff and erosion, and gives higher crop yield. Any cultivation done along the slope will accelerate gully formation, more runoff and erosion and consequently permanent damage to land. A. Planting of grasses for stabilizing bunds. Grasses prevent soil erosion and improve soil structure. The entire soil mass is penetrated by countless roots and soil aggregates and particles are enmeshed by the root system. Grasses should be grown on bunds which are not suitable for cultivation, both for checking erosion and providing pasture for cattle. Several grasses as well as legumes were tried on bunds at the Agricultural Research Station, Solapur, which receives about 600 mm of rainfall to see which of them will withstand drought conditions, give maximum root growth and canopy coverage, and stabilize bunds effectively. It was observed that Anjan planted with spacing of 15 by 15 cm produced the highest quantity of roots, followed by Marble 8, Rhodes, Thin Napier, Blue Panic, and Kusal. Legumes do not have many roots but produce better canopy within a short period, while grasses are under the process of establishment. Planting of legumes mixed with grasses is, therefore, advantageous in preventing soil erosion in initial stages. F. Planting of trees in afforestation. Forests conserve soil and water quite effectively. They not only obstruct the flow of water, but the falling leaves provide organic matter which increases the water holding capacity of the soil. If tree planting is done in the planned manner in open areas, it will serve as good windbreak, and if done along the banks of streams and rivers, it will regulate their flow. Farm forestry is another important aspect in soil and water conservation. The danger crop. G. Cashew nut plantation in coastal districts of Maharashtra. G. Farm forestry is another important aspect in soil and water conservation. If tree planting is done in the planned manner in open areas, it will serve as good windbreak, and if done along the banks of streams and rivers, it will regulate their flow. Farm forestry is another important aspect in soil and water conservation. Farm forestry is another important aspect in soil and water conservation. G. 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 Cashew nut plantation in coastal districts of Maharashtra which receive more than 1,250 mm rainfall. A new program of cashew nut plantation has been undertaken. 76. From 1963-64 on hills having slope between 10 and 20%. The sea breeze is conducive to the growth of cashew nut plants and they do not require much aftercare once they establish in the soil. Staggered trenches of 300 x 30 by 30 cm size are dug on contours at a distance of 6 meters. Cashew plants are raised in polythene bags in nurseries and 2 months old saplings are planted on the lower side of the trench with plant to plant distance of 6 meters. Package of practices of crops under rain-fed conditions. Choice of crop and varieties for rain-fed agriculture. Dry land constitutes about 75% of cultivatable lands in the country and contribute about 42% of off grains, almost all the coarse grains and 75% pulses and oilseeds. More than 90% of sorghum, pearl millet, groundnut and pulses are grown in arid and semi-arid areas. At present, the cultivable area under dry land agriculture in the state is 87% and only 13% area is under irrigation. After harvesting all available water resources, it is possible to bring it to 30%. It means 70% of the cultivable area will remain as rain-fed in the state. Under this situation, the state will have to depend upon for its major share of food production on dry land. However, the dry land agriculture suffers from two problems, viz., low productivity and high instability. The reasons for low productivity in rain-fed area are 1. Lack of moisture conservation practices. 2. Low rate of fertilizer use. 3. Lack of timely farm operations. 4. Improper crop planning as per land capability. 5. Inadequate efforts to increase water resources. 6. Unpredictable rainfall situations. 7. Lack of improved technology. The adoption of improved dryland technology will be the only answer to mitigate above situation. Considerable research efforts are being made in the state to develop the improved dryland technology since early 70s, which will help to improve the crop production in rain-fed agriculture. The important improved dryland technologies are given below. 1. Selection of efficient crops and their varieties. 2. Crop planning as per length of cropping season. 3. Developing suitable varieties for dry lands. 4. Seeding time for dryland crops. Introductory agriculture. 77. 5. Timely seeding for pest avoidance. 6. Planting pattern and plant densities. 7. Intercropping. 8. Fertilizer use in dryland. 9. Weed management. 10. Use of minimal irrigation. 11. Crop planning as per land use capability. 12. Crop planning for aberrant weather situation in dry lands. 13. Mid-season correction practices. 14. Soil and water conservation practices.